Dean of the College of IT and BT at UNIST, Professor Haedong Shim, and he will give you the welcome remarks.
Okay, good afternoon, everyone. So welcome to the International Symposium of Precision Oncology. So I'm Haedong Shim, serving as the Dean of Information and Biotechnology UNIST. So first of all, I'd like to express my thanks to all these speakers coming from Switzerland. So I heard that they are very busy persons, so it is very hard to meet them together. But this week, we have a kind of a meeting of Korean and Switzerland researchers in life science in Seoul. So we have a chance to meet all of them here today. And also, my thanks to all the speakers coming from Asan Medical Center and IBS, and of course the UNIST professors as well. Okay, so you know that during the COVID pandemic, I think that the older people may surely realize that it is quite important to nurture the professionals who is working on researching on health science and technology field. So to keep pace with this trend, the UNIST also select the health science and technology or digital medicine as one of the core research thrust. And then, as Professor Beth said, we opened the graduate school of AI this year. And then we recruit the first students last month. And also we have a very active and close collaboration with Medical School of Ulsan University. So this semester, about 40 freshman students of Medical School of Ulsan University, and then they physically stay within this UNIST campus, and then they take the courses offered by the UNIST professors. So I think this is a very unique model of collaboration between science and technology-oriented institute and then the medical school. So I think that also the international collaboration is very important in this field. So I hope that this event would be the first step for us to expand our ability in terms of international collaboration as well. So I hope that all of you enjoy the talks in this event and then share your opinions together. So it could be a good chance for making some good collaboration between various universities and medical centers as well. So thank you, all of you here today. And also especially thanks to Professor Bae and Orlando Scherer to arrange this event and invite the speakers. Okay, thank you.
Okay, next I would like to invite Dr. Jong-jae Kim from Asan Institute for Life Sciences.
Good afternoon, everyone. I'm very happy to be here with you. I think this symposium on precision oncology is surely a very historic moment for very recent and strong collaboration between UNIST, University of Ulsan, and Asan Medical Center. And I am also very happy to meet very distinguished speakers from abroad, and hoping that this symposium be a very good starting point for prolific collaboration and friendship among all the participants. Thank you very much.
Okay, last I invite Dr. Kyung Jae-myung, and yeah, he is the director of the IBS.
Okay, so previous two people even didn't prepare. So however, I'm kind of little prepared something because, and I especially I prepared this one by aiding from the ChatGPT. So I I wrote down in Korean and then ChatGPT translated for me. So, okay, so this is great. So congratulations on hosting Basic and Translation Precision Oncology International Symposium at UNIST campus. So thank you, and then congratulations to Professor Bae and Professor Schauer. So we extend a welcome to Dr. Shana Storer and Dr. Mark Rubin, who have traveled all the way from Switzerland to share important recent discoveries at this symposium. We also welcome the many physician scientists who have joined us from Asan Medical Center, a partner in UNIST Health Science Technology Program. The 21st century is expected to see the biomedical industry become one of the most crucial industries. UNIST has initiated this health science and technology program to take a leading role in this direction. It is believed that this program will produce a multitude of future talents and research that will lead the biomedical industry with the scientists and medical professionals working together. As one of the efforts in this direction, the Symposium for Health Science and Technology is expected to make a significant contribution to UNIST's leadership in the HST industry in the future. We hope that this event will be a valuable experience for both our speakers and the audience. Thank you.
Yeah, let's start. Anyway, we are quite early than scheduled, but okay, we are a little bit early, around 15 minutes earlier than we scheduled, but why don't we just start? Then we'll have enough time for discussion later.
Good afternoon, everybody. My name is Orlando Scher from the IBS Center for Genomic Integrity and also UNIST. And now I'm very happy and excited to be a member of the new HST department, and I look forward to all the opportunities that will be provided by this program for collaboration with Asan Medical School and Ulsan University, in addition to what we already do at UNIST. But very excited also to host two longtime collaborators and friends in Shana Storer and Mark Rubin. So we've been working on on a grant that's called Hijacking Transcription-Coupled Nucleotide Excision Repair for Cancer Research for several years, and maybe I tell you just 2 words about this program. So this is a Synergia grant. This is a grant by the Swiss National Science Foundation for national or international collaboration. And so it's been my great pleasure to work on this with them. And then the other thing that was already mentioned is that the Swiss Embassy is hosting the Swiss-Korean International Science Week this week and a symposium. And the Swiss Embassy is very active in fostering collaborations between Korea and Switzerland. And so with the spirit also in Korea now of having more international collaborations, I encourage many of you to sort of reach out to see if there's any programs there that might be of interest, and I'd be very happy to tell you more about these. So for example, one of Shana Storer's students, Claudia Aloisi, was in our laboratory for a month a couple of years ago on an exchange fellowship through the Swiss Embassy. So I hope you all can take some opportunities to do that as well. Okay, so I also welcome everybody here to the symposium, and I hope it will help us connect with UNIST and Asan Medical Center. And also special welcome to the students, even though you seem to be seated a bit in the back. As always, I encourage you to ask the questions and engage in the discussion, and hope to have many contributions from you as well. Okay, so just one last word on the international collaboration that I have with Shana and Mark. So I'm Swiss and I'm in Korea, and Shana and Mark are American and they're in Switzerland. And so that's our Swiss-Korean collaborations, Americans in Switzerland and a Swiss in Korea. So I hope you can take opportunities like that as well, that you work internationally with people no matter where they're from or where they reside. All right, with that, I want to turn to introduce our first keynote speaker, Shauna Sturla. So Shauna is currently at the ETH in Zurich, where she's the professor of toxicology. She's originally from Brooklyn, New York, and did her undergraduate work at the University of Berkeley, and then a PhD at MIT with Steve Buchwald on organometallic catalysis, so a hardcore chemist. So for those of you who don't know Steve Buchwald, he's one of the pioneers in making new reaction methodology to synthesize complex organic molecules. After that, Shauna moved to the University of Minnesota, where she first did a postdoc with Steve Hecht. Steve Hecht is a pioneer in carcinogenesis and DNA damage related to smoking in particular. And then she started a faculty position at Minnesota, rising through the ranks, and then moved to Switzerland about 15 years ago almost, I guess, right? Where she has been ever since at the ETH. And she runs a very broad interdisciplinary program that combines chemistry, bioanalytical chemistry, and also biology to look at various aspects of toxicology with a focus on DNA damage and repair. And so this is how we've known each other for a very long time. And so her work has looked at both types of DNA damage, at DNA damage detection, looking at the biological consequences of DNA damage, looking also how drugs or natural products are bioactivated to form DNA damage. And more recently, she has had a very successful program on DNA damage sequencing, that is, finding ways to look at where in the genome particular types of DNA damages are formed and located. And I believe that is what you will be telling us about today. So Mark and Shauna, welcome again. It's a great pleasure to have you here, and we look forward to the presentations.
Hello, can you hear me well? Yes? All right, good afternoon. Thank you, Orlando, for this lovely introduction. It's a real treat being introduced by somebody who knows me really well. I think maybe I could keep Orlando up here to continue and give the talk. I say it as a joke, but as you see slides come up later, it's not so much of a joke. But this is my first time in Korea, and I'm having a really lovely visit. I'm really happy for the opportunity to be here, and pleased to share with you our research at the ETH Zurich. So you see already up here on the corner is the name of the department that I am part of at the ETH, which is the Department of Health Science and Technology. So I'm glad to represent the ETH Department of Health Science and Technology here for this kickoff event and all of the things looking into the future. So in our department at the ETH, we have a diverse mix of labs that are representing areas of basic science, as well as engineering and technology, with the idea that all of the basic research that is happening in the department is then connected to translational applications. And we also have education programs, 3 education programs within the department, which is students who are studying a major in health science and technology, a major in food sciences and nutrition, and also the medical curriculum is in our department, so a bachelor's in medicine. So I look forward to future potential connections between the 2 departments. So let's see if we can get past this slide. It's this. Yeah, got it. So I will start with unraveling the genome from human cells, which is a focus of interest. So in cells, on our chromosomes are raveled what then comprises the bases of DNA, so the 3 billion base pairs in DNA. Most of this genetic information is actually quite similar amongst many of us, and these coding for these 21,000 genes, there's just actually a very small portion that defines the differences between individuals and also contributes to disease processes that are relevant, for example, in cancer. And we're particularly interested in the interactions between the environment and our genes. And so DNA continuously undergoes chemical modifications. This can be due to endogenous factors, such as metabolic processes, spontaneous hydrolysis of DNA bases. We're also interested in metabolic processes that happen, that are catalyzed by bacteria in the gut microbiome that can generate reactive species. And then exogenous factors like the use of tobacco, UV irradiation, pollution, exposure to chemicals in foods as well as drugs can then lead to damage in the DNA. About 100,000 damage events happen per cell per day, and these are comprised of breaks, gaps, abasic sites, alkylation adducts, oxidation products like adeoxoguanine, cross-links, photodimers, and deamination, etc. So what are the cellular consequences of these DNA, these chemical reactions basically happening on the DNA? This can either have an adverse consequence, but this can also have a therapeutic relevance. So, for example, the DNA damage, if this damage is arising from a cancer drug, the DNA damage can be repaired by DNA repair. This can also be then a substrate for error-prone replication, and this can lead to mutagenesis, or this can lead to drug resistance. Again, if that damage comes from a cancer drug, if this evades DNA repair and inhibits DNA synthesis, then this is actually a beneficial aspect of cell death. And this is extremely therapeutically useful. So the oldest anti-cancer drugs and some of the most widely and effectively used anti-cancer drugs are really based on this process. And so genomic alterations that result from anti-cancer drugs is a basis of potentially tracking precision responses to alkylating agents. And so we can use this not only for developing the drugs themselves, but as a therapeutic basis of how an individual cancer responds to certain drug exposure. And as Orlando alluded to, we've been very interested in answering the question of how the location of the location in the genome where a reaction is occurring, how does this influence the biological impacts, the cellular impacts of interaction with the DNA damaging agent? And in this context, we have been working on developing a variety of strategies for profiling drug and chemical-induced DNA adducts. So a DNA adduct is the chemical product of the reaction of the chemical or drug with a base in DNA. I've got the little red ball attached to a guanine because guanine's a typical site of modification. And if we look at this plot of mutations, representation of mutations in the whole genome, we are outstanding at understanding what that landscape of mutations is in a cancer genome. So we can really track this using modern DNA sequencing technologies extremely well. But we are not very good at tracking the precursors of those mutations, so these products of these chemical reactions that can give rise to adducts. We're not so good at tracking those in the genome. And that is what we are working on, trying to address and trying to understand what is the distribution of these adducts, how that distribution is influenced by the damage tolerance network as a basis of mutational signatures that can arise in cancer. And so mutational signatures, as illustrated with an example here, shows the relative frequency that each of the 4 nucleotides in the genome is mutated in each of the 96 possible trinucleotide contexts in the genome. And so we can look at where, in terms of what genes mutations are on, but then we can also look throughout the genome as what is the frequency of those mutations. And you'll hear a lot more about this in the next talk or the one after that by Dimitri. And then we can understand how this contributes to mutational signatures that arise in cancers that are have an etiology in certain chemical exposures. And then you remember in my first slides, I was talking about this as a basis of how cancer drugs induce cell death as well. So I want to talk to you about both chemistries that drive the development of cancer, as well as chemical alkylation as a basis of treating cancer. And this gap that I mentioned that we're not very good at yet is then linking what is the DNA damage signature that is the potential precursor of these mutational signatures. Okay, so I will show you 3 examples related to this general context. I'll start out by talking about DNA oxidation, which is one of the most prevalent forms of DNA damage. And then I'll give an example of alkylation by a tobacco carcinogen or by a cancer drug. And so we start out here with a main oxidation product, which is 8-oxoguanine. And 8-oxoguanine can arise from the formation of reactive oxygen species that can result from chemical exposures as well as from endogenous metabolic processes that increase oxidative stress. Of course, our cell has an exquisite system to buffer the formation of oxidative stress, but this can get overwhelmed by exposures, and this can give rise to mutations. And so we wanted to ask the questions of how much of the genome is oxidized and where. What are the locations of those oxidation products? So I go back to this picture of the genome that I started out with, and there are strategies for measuring oxidative damage that we use very much in the lab that are extremely useful, that don't give us any location information, but tell us a lot about how much is there, and under different conditions we can see how the oxidation increases or is mitigated. So assays like the COMET assay and using mass spectrometry are very useful for that. And we can start to increase the resolution of these assays. And there are a couple of, for oxidation, you have here a couple of examples of getting higher resolution methods. But when we started working in this area, there was no way to map with single nucleotide resolution where these oxidation processes were happening. And if I really want to address this goal of generating DNA damage signatures, in analogy to the dimensions that we look at for mutational signatures, I really need a way to map the damage at single nucleotide resolution. And so we have developed a couple of different strategies for mapping DNA damage in general, and in particular for mapping oxidative damage. We utilize a strategy that takes advantage of an enzyme, so a glycosylase enzyme that recognizes oxidized bases in the genome, removes the oxidized bases, and then at the gap position we can ligate an adapter sequence that has a reference sequence on it, and then by sequencing we can find where the damage locations are. Another strategy I'll talk about later is more analogous to ChIP-seq approaches and uses an antibody pull down as a first step. So in more detail, the way this method works, which we call click code seq, is that glycosylase, so adeoxoguanine glycosylase, is used to excise the oxidized base. We incorporate an alkyne substituted DGTP analog, and so we have a 3' alkyne group that is incorporated there. We take advantage of click chemistry with an azide, 5' azide terminated DNA strand, and this DNA portion has basically a reference or barcode sequence that we then can create a linkage into the DNA, and then this triazole is bypassed by polymerases, just like a phosphate, and so we can take advantage of that to do standard library preparation for DNA sequencing. And in the library preparation, we know that wherever our orange reference sequence ends, that this was the location of an oxidized base. Okay, so here is here is just a snapshot of then addressing whether that oxidative damage is distributed evenly throughout different aspects of the genome. So we can look at all sorts of things that are annotated by other DNA sequencing approaches. So we can look, for example, at transition start sites and termination sites. We can look in telomeres. We can compare with RNA Pol II occupancy. And you see a large dynamic range of differences, so a lack of homogeneity in how the oxidation is distributed throughout the genome. The data that is shown right here is data that reflects the background distribution of ADOXO-G in a yeast genome, which is where we started when we were working on this project. I will just show a couple of more pieces of data from the yeast genome to keep things a little bit simple, and then I'll show you how we have moved on to human genomes. So one thing we can then look at is this flanking base identity, which is the basis of the cancer genome mutational signatures and how this impacts adeoxoguanine distribution. So we can see a correlation between the ionization potentials of the different triads in DNA and the propensity to oxidize. And then these data, also all three data on this slide are different representations of the same concept. So looking at what is the relative frequency in different sequence contexts, and we see that we have large differences driving that. And so from this, we could see we can see that the global genome architecture and protein binding can impact the damage distribution, and we developed this nice click code seq method. Despite our implementation of different blocking protocols in the technical preparation of these samples, a main limitation that we found here is that we have a lot of background damage. So the way the glycosylase is working is generating a gap in the DNA, but we have a lot of gaps that we're competing with in the background of the DNA. We have AP sites that we're competing with. And so we're trying to map this number of ADOXO-G sites, but we're trying to do that in competition with these kinds of levels of background damage sites. And so what we learned from working with the yeast genomes was really the importance of really blocking the background damage. And so we then went on to spend a lot of time refining the experimental protocols. We had to develop new data analysis workflows to map oxidative damage in the human genome. And so there are a lot of different strategies that we can use to block and repair these breaks in DNA. And as a basis for optimizing how to do the initial DNA preparation for that work, we developed a fluorescence-based method to track how much of this alkylated nucleoside analog we were putting into the DNA. And so this is the same as the click code seq method that I showed you the sequence of, except now instead of clicking in a barcode sequence and then sending it for sequencing to get our data, what we do is we just click in a fluorophore. And so this is a fluorophore that is activated when we form the triazole. And so that we can do this very quickly and very easily, and we can track how much of the triazole is forming just simply based on fluorescence. And we see that it has a really nice quantitative response, and so this is a really convenient way to track this mapping that is happening in the genome, and this was really important for us to optimize our blocking methods so that we can test many different conditions of enzymes that we use to process the DNA, how that influences the levels of fluorescence we had as a basis for what sites we are actually able to label in the DNA. So, okay, so I have here version 2.0, version 3.0, et cetera. So there was a lot of optimization, but now we can generate for the first time low background oxidative DNA damage signatures in human cells. So the data I show you here is for HAP1 cells. You have an overall perspective here of the damage distribution. We have some, these high values that you see here are not related to overabundance of oxidation there, but more related to difficulties in assigning specific, let's say, specific sequence contexts in these locations. And so we can look at this on the DNA damage signature context. So in the dashed lines, this is the background genome context. So the number of times you have this sequence context in the genome. What you see in yellow is the damage that we see. This is just the background oxidation. Cells grown in cell culture, we always have typically have a background oxidation signature, and so what we see here is that, and it's very similar to the SBS signature 18, which is attributed to reactive oxygen species. And so it was very affirming to us that this matches quite well to the oxidation signature that we generate using this method. We made some other interesting observations in terms of how the oxidation is distributed in the cells. So it seems that strikingly, ADOXO-G is underrepresented in the CPG context. So this suggests that at CPG sites, the oxidized guanine is influencing the formation or the formation of methylated cytosines, or the methylation process of those cytosines is somehow influencing the repair of the oxidized position. And we also see that in the promoter regions of some expressed genes, we have an enrichment in ADOXO-G. So we can do this, for example, by comparing to DNA hypersensitivity sites, and this gives a sense of how open the genomic regions are. And then finally, we see an interesting relationship between the strand bias of the oxidized bases with gene expression. And so we can divide the gene expression data into going from not expressed to highly expressed genes, and we can see that we have a clear increase then in the strand bias. And that's the interpretation of this data is shown in this cartoon, is that you have a increased maintenance of the ADOXO-G in the transcribed strand of these actively expressed genes. So this shows a really interesting link for us between oxidation and transcription. potentially with a transcription-coupled relationship with ADOXOG, which we didn't necessarily expect. So the conclusions I can make with this data from the human cells and ADOXOG is that we newly optimized a version of ClickCode-seq that enables high-resolution oxidation mapping in the human genome. And so our original method, that we'd used in yeast, conceptually it is the same approach, but from an experimental perspective it's really quite different because we can't do the same process that works in yeast. It just doesn't hold up for the human genome. And we see this anti-correlation at CPG sites. I didn't show data with chromatin features, but there are some relationships we're exploring there. And then we saw this interesting strand asymmetry with gene expression. Okay, so that was a focus on oxidative damage, and I have on this slide three images from three publications from the lab. One is a review article, which is called Next Generation DNA Damage Sequencing, and it covers all of the review is actually, it's 2020, so it's 3 years old, so it's like totally out of date now because the field in the last couple of years has really moved forward with a lot of new methods for DNA damage mapping approaches. And in this review article, we cover all of the methods that were developed up until 2020, what the basic approach was, and what kinds of damage products could be mapped. So also, for example, photodimers and platinum adducts. The middle one is something I won't get to talk too much about today, but well, I won't get to talk about it all today. But this is a carboxymethyl DNA adduct that seems to be elevated in individuals who have high meat content in their diet, and so we're interested in understanding how this is mapped in the human genome and how that tracks with colorectal cancer risk. And then on the right here is the benzoate-pyrene adduct that results from carcinogen benzoate-pyrene, which is a bulky adduct that results from tobacco exposure. And so I want to tell you then about this as a real contrast to the small oxidation damage that I talked about in the last example. I'll go now into benzoate-pyrene map, which is this study is a first comparison between how quantities of alkylation track with changes in the location of these adducts in the DNA. So really a first DNA alkylation map in the human genome. And before I start there, I just want to disclose that some portions of the work that we did on this tobacco carcinogen were funded from Philip Morris, which is a tobacco company. So the strategy that we use for profiling the alkylation adducts is based on the conceptual approach that was developed in Sankar's lab and has been used for other bulky adducts. He calls this high sensitivity damage sequencing. And the concept here is that the DNA is fragmented and then you do an initial pull down with an antibody that is directed toward the DNA adduct. And then a high fidelity polymerase is used to replicate using those fragments as templates. And so the high fidelity polymerase stalls at the sites of these bulky adducts. And so we adapted this approach to do this with benzoate-pyrene. And this took some modifications, obviously, in terms of the antibody, as well as the verifying the stalling of the polymerase by the benzoate-pyrene, by the N2 BPDE adduct at DG, which is a major benzoate pyrene adduct. And then in this study, we looked at both using mass spectrometry to quantify increases in the adduct, and then we used this antibody-based localization strategy for sequencing. And then we looked into the data to find what is the similarity of these profiles to lung cancer patients. And so what we can see on the quantification side is an increase in benzoate pyrene DNA adducts with increasing exposure to BPDE. And then we did this both from cells exposed to BPDE and naked DNA. And we did the naked DNA to use this as a control that if the DNA is not assembled, we expect that we have a more random distribution of the damage. And then we looked for regions in the cell that either have an increase or a decrease relative to the naked DNA. And here again with the whole genome view on that, and then we can zoom in, and here is a focus on a 10 megabase region of chromosome 2, and what you see is this plus-minus association. So at each of the locations, where do you have either an increase or a decrease in abundance relative to the naked DNA, and this relationship is going with increasing concentration. And so in general, when we look at, as you get into these higher levels of the BPD exposure, you see a pretty similar profile in terms of the regions that are modified. So this was actually a little bit surprising to us, because we thought that as we went up in concentration, that we would start to really saturate the genome, and that we would lose, we expected to see some sort of selectivity profile in the low concentration regime that we would lose and be saturated. And we found that it was actually surprisingly consistent which of the regions were prone to be alkylated. And so, then here is another perspective on the same data. So I showed it there more kind of in a meta-profile sense. And here we can see what were certain regions that had either an underabundance or an overabundance of the modification. And then we can look at that again with meta-profiles related to regions, for example, before the transcription start sites that have increased levels of alkylation. And now we can look at that data was in terms of what regions of the genome, what regions of the genome does the alkylation accumulate in. And so because we're working with single nucleotide resolution data, we can extract a DNA alkylation signature, so a damage signature. And this is the trinucleotide enrichment at the alkylation site, and so this is the background of the unexposed cells. And you see here is the cells exposed to BPDE. I didn't mention what the cells were at the beginning, but these are BES2B cells, which are, these are normal human lung cells. So, so we could extract signatures using non-negative matrix factorization, so the same approaches as we use with mutation signature analysis, but we're using this for the damage signature, so focusing on these modified, the frequency of this modified guanine. And we see we could extract two different signatures, signature A, which seems to be background signature, and signature B, which has this very high cosine similarity to the profile of the characteristic C to A mutations in the SBS mutational signature 4. So this is from the cosmic somatic mutations, and this is the mutational signature that's prevalent in lung cancer patients. We can also look at with the different in the different samples the relative contribution of the experimental signature. Of the extracted signature to the profile that we see in the sample. and we can then we can look at regions, we look in these signature contexts, and then we could also also kind of surprising to us is that with this data we were actually able to look into hotspots we were able to look at hotspots in specific genes in the genome. And so to guide this, we looked at prevalent mutations that are identified in lung carcinomas. So these are looking at frequently mutated sites for a large number of patient samples, so publicly available mutation data. And I'm showing here two of the examples of some genes that we found. So the CDK and the KEAP1 gene, and you can see the star indicates that we located an alkylation site at the particular concentration at this particular location. And you see, we don't see it, we're not able to see them at the lower concentrations, but we get a pretty robust signal then going at the higher concentration. This site that we found, the modifications, or these two sites are the most frequently, for these genes, are the most frequently mutated site of this gene in lung adenocarcinomas. And of the several thousands mutational sites that are characterized in lung cancers. This is the 28th most common. I guess it's referring to, I'm not sure which one is the 28th most common, the CDK or the KEAP1. I have to check that. But then we also found alkylation in other prevalent hotspots, but we found that less frequently, and we didn't find the same kind of robust correlation of having them form at the higher levels of concentration. Okay, so with this DNA alkylation, this is the first single nucleotide resolution map of DNA alkylation in the human genome, and while the alkylation levels increase with increasing chemical concentration, as we would expect, surprisingly, the enriched areas of the genome are actually quite consistent. These tend to be the compacted regions of the genome, and the interpretation there is that the compacted regions of the genome are then inaccessible to repair. And these regions that have low alkylation persistence are those regions that are actively prepared. And we haven't tested that. We haven't tested that in this data set because we are just looking at repair proficient cells at a particular time point. I think this is after about 24 hours of exposure. And so we know that repair is happening and we're not controlling for the repair. And so it would be really interesting to look at the dynamics of that process. We've done a little bit of that. We do the same. I'm not going to show data for UV here, but we've also done a lot of studies with UV damage mapping using the same strategy. And in that case, because with the UV damage, we can do a really controlled, of course, initial exposure to the UV damage. This is a really clean way to track the dynamics with the, in this case, with benzoate pyrene, you have this complication of the metabolic activation that is needed for the DNA alkylation, and you have competing rates of metabolism. You see that we did these cell exposure studies actually with the activated form of the chemical to try to control that a little, but it's not as clean as the UV. And so we're really looking into these dynamic questions, focusing on UV damage instead. So that will be interesting to see how that working model holds up. And then the benzoate pyrene signature has a highly similar profile to the mutational signature that is prominent in lung cancers, and we can even get that down to a genetic mutation hotspot level, and we can find that these common mutations are also frequently alkylated. So this is a summary of the two examples. I showed, and we can extend these strategies for other modifications, chemistries, other exposures, and different projects in the lab are then looking at different kinds of modifications. We look at metabolites from the gut microbiome, damage that is linked to the diet, and we're also interested, as I alluded to at the introduction of my talk and linked to the oncology topic of the symposium, for DNA damaging drugs. And the one I've shown here is temozolomide, which there's also an interest in this institute in temozolomide. And what I would like to, the data that I'd like to share with you today is on temozolomide just delivers a methyl group to the DNA. And this molecule, trabectedin, or ET743, is a really structurally interesting molecule that results in the formation of a bulky DNA adduct that is shown in this nice animation. And so this is from, it's a natural product derived from a sea squirt, and this is being developed as an anti-cancer drug. This is an interesting observation by, this is data from Visesato Moore, who is at the institute here. And what you see here is the relative sensitivity to trabectedin of cells that have different nucleotide excision repair factors lost. And you can see that the CSB knockouts are very resistant to trabectedin. And I mentioned at the beginning, I said it was no joke about having Orlando come up here and finish my talk. And so if some of you have seen Orlando give a talk before, you'll recognize this mechanism for nucleotide excision repair, which is it's like the world famous figure because of Orlando, and showing the NER mechanism. And what I really want to highlight is that there are two important pathways for nucleotide excision repair that differ in the recognition of the DNA damage. And so we have global genome nucleotide excision repair and transcription-coupled nucleotide excision repair. I alluded to the TCNER pathway when we were looking at the 8-OXO-G data that had the transcriptional strand bias. And this is initiated by a stalling of RNA polymerase II and a recruitment of CSB. And this is and so this allows us, these CSB proficient and deficient cells really are a nice way to differentiate between sensitivity of drugs that are being influenced by the TCNER subpathway or the GGNER subpathway. And then the subsequent processes are the same for the two. So we have the sequential incision, resynthesis, and ligation. And so and so I guess when I mentioned when I was talking about the benzoate-pyrene adduct, and I was talking about the accessibility for repair, and I compared this then with the UV data, as I think many of the people in the audience who are interested in NER realize that I was really referring to NER processing of those benzoate-pyrene adducts. But okay, so this is really interesting to see the CSB resistance profile, and this suggests that the drug is actually working very well in cells that are TCNER proficient. And this is actually an interesting basis of how many years ago Orlando and I started collaborating, is because I have worked for a long time on a different molecule that is called acylfulvene, and this molecule seems to be uniquely repaired by the TCNER subpathway. So what we see is really an opposite profile of selectivity, and we're very interested to understand what controls that selectivity and how we can basically, as Orlando said at the beginning, hijack the process so that we can really preserve the strand breaks that are generated by NER as a basis for inducing cytotoxicity in the cancer cells as a therapeutic basis. Okay, so the hypothesis then that we are working on is that the trabectedin adduct blocks this second excision during repair, and that this is a TCNER dependent process. So this is recognized. XPF can make the first incision, but then the second XPG catalyzed excision is blocked. And so we wanted to ask the question of what are the genomic characteristics that then impact the distribution of these breaks that are induced throughout cancer genomes, and what is the relationship with TCNER. So, so the theme was the damage mapping methods, and this we used another one here, which is actually quite similar. It's a quite similar concept of what I explained with the ADOX-OG, except in this case we are adding this a proximal adapter to the location of the strand break. So we're looking at the 3' OH ends of the DNA, and so we get here to start with. We don't have to use a glycosylase like we do with the ADOX-OG, and then this is biotinylated. We can capture these strands and then identify the locations of the strand break based on the proximity to the adapter sequence. And this, applying this and the use of this method for the 3' strand breaks was a really developed as a really robust method by Helle Ulrich's lab, and previously published. And in collaboration then, between all of our labs, we have worked on an upgrade of this GLOWY-seq method, and we're now changing it in two ways. One is that we're really using the trabectedin to induce the strand breaks, so we're looking at this drug-induced formation of the strand breaks, and we're also adding an indexing, a distal adapter that has an indexing sequence. So this allows us to distinguish, basically add a way to count how many of the biotinylated proximal adapter sites we have for each of the distal adapter indexes. And this gives us a way to quantify in different cells where the modification is, and really allow us to not just say that we have a modification at a particular site, but if we look at a population of cells, we can look at the relative frequency at those sites. So this is a really nice quantitative upgrade of the method, and then we call this this new approach TrabiSeq, and we've used that to study the strand breaks that are induced by trabectedin. And this is again, this is now the results of the collaboration that Orlando was mentioning at the beginning, and we have I have some data where we can look at, for example, a portion of chromosome 19 here, and we have on the top the cells where TCNER is active, on the bottom TCNER is inactive, and you see the difference between the blanks and the treatment with trabectedin. We have a lot more strand breaks generated in the trabectedin, and then this is kind of a same overview where you see when you really require the activity of the TCNER to give you all of these strand breaks throughout the genome. And so when we looked at these two perspectives, I said to our postdoc, Vakil Takaviv, who's working on this, we were really excited to see the increase, but I said, well, if we would have done this without anything sequencing based, we would kind of have the same answer. So what is it that we learn from using this in a sequencing based way? And so there's actually a lot of new things that we have learned from having this on a sequencing level. For the sake of time, I'm just going to highlight one very interesting relationship we have seen, which has to do with transcription, and we see that these DNA breaks accumulate in the transcribed strands of genes. And so here we have, again, like we did with the ADOX-OG, tiers of gene expression, and you can see this bias between the transcribed strand and the non-transcribed strand, and the difference between the TCNER activity and inactivity. And we can also, we can look within the genes, and then we can also look in that this is really focused in sub-regions of the genes. And so we see that when TCNER is active, we have really early relative to the transcription start site, we see this flipping in where the strand breaks accumulate. And so this shows an interesting relationship then between, basically we uncovered that the breaks map to the transcribed genes of, the transcribed strands of active genes. And then basically this is a new strategy then for profiling cells that are responsive to the drug and as a mechanistic probe for nucleotide excision repair. So with that, I'm going to wrap up. You can see in these 3 examples I showed today how we're using genome-wide DNA damage patterns and sequence-specific localization. These are new methods that are telling us what is the chemistry and what is the damage that is happening in DNA, where is that, how much of it, and we combine that with mechanistic studies to understand how that influences cellular processes like mutagenesis or cytotoxicity, and we can use this information to understand who is at risk from mutations arising or who can benefit from cancer therapeutics. So with that, I thank the group in Switzerland who did this work, a special acknowledgement to postdoc Vakil Takaviv, who I already mentioned, who is working to on this Synergia project that Orlando told you about. Nico and Navnit are other PhD students in the lab who are focusing on the oxidation damage work. And also we have a collaborator in Dresden, Anna Poch, who is an expert on oxidative damage. Former members of the lab who contributed mostly to the alkylation mapping work, funding agencies, and yeah, thank you for your attention.
Okay, great. Thank you very much, Shauna, for this wonderful overview of DNA damage sequencing in various contexts. We have time for some questions. I'm going to start back here with the students to have any questions that you might have. Anybody?
Thank you for your interesting presentation, Professor Surla. During your presentation, I imagined that the timing of the sequencing with the different techniques would result in the different data. So I was wondering whether you have tested the repair kinetics by changing the sequencing timings.
Yeah, I think this is a really important question, and it's what we're very much in the middle of working on right now. It's what we're in the middle of working on right now, and it's actually it's a little trickier than it may seem because the kinetics are always an addition. You always have a profile of the formation overlaid on top of a profile of the removal, and so for the for the alkylation damage, one of the ways we're trying to address this is by looking at UV induced damage so that we can really turn on and off the UV, induce the damage at an initial time point, and then track that over time. So I don't have the data, but exactly what you are suggesting is what we're working on. And we're also right now trying to do the basic kinetics with the oxidation in the cell models from a quantitative perspective first to really understand what are the time points that we need to do the mapping to try to get at what you're saying. So really important point, and I hope that I'll have data soon to give insight.
So the question about the benzopyrene mutation signature. So it's known that POL-kappa can bypass benzopyrene adduct efficiently. So what polymerase do you think is contributing to introduce benzopyrene mutation signature?
You know, I always think of Pol-eta in bypassing benzoyl but I think of this from biochemical studies that don't involve mutational signatures. So I haven't checked that in the mutation signature. Maybe Dimitri has an answer. Dimitri and Tony might have an answer to this. Yeah, yeah. I'm gonna guess Pol-eta, but to be checked. Actually, not just. I mean, potentially not just Pol-eta, but then the combination of Pol-eta and Pol-zeta as an extender.
Yes. And do you think this benzopyrene-mediated mutation signature is associated with lung cancer development? So maybe benzopyrene is known to be repaired by nucleotide excision repair. So I don't know whether XP patient, maybe the patient whom is smoking, develop a more cancer, a lung cancer.
Yeah, I mean, maybe if I could phrase it, I'll phrase your question a little bit more generally. Is maybe what you're suggesting is really the use of the mutational signatures as a diagnostic basis of predicting cancer risk. So you can think about that in many different contexts and how that could influence somebody's behaviors, or this is one where maybe somebody can make a decision to stop smoking, but there could be other situations where maybe there are interventions that you could look into developing interventions to protect certain people who are at risk, and you can use the evolution of the mutational signatures on one hand, but really the damage mapping as showing the precursors of the mutation as a preventative diagnostic strategy. So I like this.
Of course, there's many questions, but for trabectedin, there's the enrichment at the transcriptional start site. So that can be seen in two ways. Chromatin is open, and therefore you have a modification at the site where chromatin is open, or in the context of transcription coupled repair, that you have a modification somewhere in the gene body, as then repair is blocked in a toxic intermediate. You actually do something with transcription coupled repair that reverts back to the start site. Mm-hmm. So a more indirect model.
But it's more indirect, but it's both. They're both, in a way, they're both related to the same foundational phenomenon.
Yeah.
I guess because of the accessibility, like the openness because of the active transcription is then related to the NER recruitment.
Right.
Right?
Well, we might have surprises, I guess, but I don't know. But the idea still is that trabectedin still, as an adduct, somehow binds to XPG and inhibits.
Yes, this is still that it somehow binds to XPG, but we're not, with this data we can't directly probe that with this data. So this model, so that this kind of doesn't add to or remove from this model of XPG binding.
Okay.
So I can't, I can agree with that statement just based on the older data.
And then maybe just final question. So what is the hottest hotspot in kind of molar term? You know, how many moles of bases are modified versus unmodified? Is it a 1 in a million, 1 in a...
You mean in terms of the density?
Yeah,
yeah. Like how many...
You know, if you counted 10 million molecules, what would be, how many would be modified
yeah,
in the, you know, CDK example?
I'll have to look into it because each of the counts we're doing are really small.
Yeah, I know.
Yeah. So it's usually, I mean, we're looking, you know, like exactly this with the CDK data. You know, we're looking at 1, we're looking at 0, 1, 2.
Right. And in a way, in most cases, it appears that the primary damage sequencing and the signatures are very strongly correlated, right? Certainly, that there's a strong correlation between primary damage and mutation, at least for the 8-oxyguanine.
Yes. Yeah. Go ahead. Keep answering. I have many thoughts on this.
Yeah. So the answer is yes.
Yeah. But you're asking. I think what you're asking is because I think a lot about the contribution of damage and the contribution of repair. But this gets back to the first question. That was asked, which is like the alkylation signature we're looking at I think is really a repair persistence signature, not a formation signature. So so it's so I think I would say what relates to the end product signature is the persistence, not necessarily the formation. And there might be contexts where that is driven strongly by formation, but like in the benzoate-pyrene example, I would say that data is saying that it's driven by persistence. But we need the results of the experiment he suggested to really say that.
Okay, thanks.
Okay. Other questions? I have one. It's interesting to see the extra 8-oxo-G in the CpG sequences, right? So I was wondering what that might, I wonder what this might be attributed to. I would imagine in chromatin context, 8-oxo-G you form no matter what the context is in chromatin, right? Formation may be relatively unimpinged by that. So do you think it is because chromatin dynamics induces faster repair in those sites, or is it a sequence context where something special happens at CpGI?
I think it's the first. I think it's the first, and I think that would be consistent with some other data that is looking at the mechanism of the CpG maintenance, and that this opening process promotes the accessibility. But it is possible that there's actually some proximity effect of how easy it is to oxidize. But I think there's so much strong evidence from the CpG the CpG methylation mechanistic work that suggests the first model.
Okay. Thank you very much again, Shana, for coming all the way to share your work with us. All right, and so we'll move on to the next speaker, which is Dmitry Ivanov from IBS CGI, who will continue on this theme of DNA damage sequencing, but now look at the actual mutation signature. So what's the longer term consequences of this damage? Dmitry.
I would like to thank the organizers for giving me a chance to present our work here. And this project that I'm going to talk about is a result of close collaboration between IBS and UNIST. And already talking in the morning with Professor Sturla, we found a lot in common, and maybe it's a start of more collaboration with Switzerland as well. So, genome is under constant attack from both endogenous and exogenous DNA damaging factors. And all this damage is either reversed by DNA repair pathways or converted to mutations. So there was an idea that mutagens leave footprints of sorts on genomic DNA, and using a comparison which was first introduced by Professor Nick Zainal, looking at these footprints which mutagens leave on genomic DNA is not unlike looking at the traces that humans left while walking on the beach. So by looking at these footprints, one can possibly deduce the history of the cell exposed to genotoxic agents. And this is pretty much what we are trying to do. By looking at mutational signatures, we are trying to deduce the history of exposure to genotoxins and the origins of cancer. So what is mutational signature? As Professor Sturla already introduced, all mutations were classified originally in Mike Stratton's lab depending on the preceding and the following nucleotide. Thus, there are 96 classes of mutations. And this is the signature 1B, which I am showing here. This is a signature associated with aging. So when 5-methyl cytosines get spontaneously deaminated, they get converted into thymines. Therefore, this aging signature is mostly C2Ts at CG sequences. And then this is the single base substitution signature. This information about single base substitutions is further supplemented by information about indels, small insertion deletions, as well as large structural variants like larger deletions, duplications, inversions, and translocations. The first signatures were computationally derived from cancer genomes in Stratton's lab. But the problem with this pure computational approach is that usually several signatures can be derived from one genome, and one doesn't really know when to stop deriving them. And only in very few examples it was obvious what was the cause of these signatures. Like these examples, for example, were UV exposure and tobacco smoking. But in many cases, the signatures, the origins of these signatures remained enigmatic, and therefore it was very helpful to start obtaining experimental signatures from defined DNA repair mutants and mutagen-treated cells, so that these experimental signatures can then be compared to computationally derived signatures, and then one can draw the conclusion about the origin of cancer. So this is exactly what we set out to do in our lab at IBS. We use in our experiments TK6 cell line, which was developed by Professor Shunichi Takeda at Kyoto University. And he also generously provided us with his collection of more than 120 DNA gene repair knockout mutants. The good features about this cell line are that they are almost wild type. They have very stable karyotype. They hardly ever misaggregate chromosomes. They are P53 proficient, and they are semi-diploid. And then we also occasionally use, as I will show later, HAP1 cell line, which was used previously in this whole genome sequencing mutational signatures papers. So in our experiments, we have to deal with clonal populations, because if you try to sequence a mixture of cells, then the mutations will be essentially diluted with non-mutated sequences, and one wouldn't be able to make sense of it. So we start growing a clone from a single cell, and then at some point we divide it into 3 portions, expose it to different concentrations of the genotoxic agent to leave untreated, and then we do the second subcloning step, during which stage the mutations get amplified. And then we sequence 3 clones per condition. And what we do is we only look at mutations which are present in one clone but not in others. So all the mutations that we study were generated in the course of our experiment. And this is an example of what kind of mutations we can be looking at. So for example, mutation 1 was generated during this growth from a single cell, the first mutational accumulation growth. And then this mutation would be present in some clones but not in others, and we would score it in our analysis. Mutation 2 was generated during mutagen exposure, so we will have one clone which has it and two which don't. And mutation number 3 appeared very late during this mutation amplification growth, and it will be present only in a small fraction of cells and therefore will be disregarded in our analysis. So the first question which we decided to study is what is the mutational background of the cell line that we study? And it somehow sounds like a boring question, but actually if you look at the background, there is a lot of interesting information contained there. There is a recent paper from Martin Carena's lab where the researchers went to the zoo and they isolated natural clonal populations from intestinal crypts of different mammals. And what they found is that there was a very strong correlation between mean mutation rate per year and the lifespan of an animal. So the animals which live very short lives, like mice and rats, have very high mutation rate, and humans which live long lives have very low. And then when they looked at what kind of mutational signatures are actually generated in these intestinal crypts of different animals, they could see that there are basically 3 components present. One component is that SBS1 aging signature, which is spontaneous deamination of 5-methylcytosines, which I already introduced. Another one is what Professor Sturla was talking about, is the oxidation signature SBS18, which they see as a component C. And interestingly, this oxidation signature is actually very prevalent in animals which run around a lot, like mice and rats, and is practically absent in animals which spend most of their lives sleeping, like lions and tigers. And then there is a mysterious component B, which is signature 5. And the etiology of this signature was unknown until basically this year. And the signature 5 now is considered some kind of a kitchen sink of a signature. It probably is a combination of several ones, but one of which is signature SBS40, which is a new generation of this component signature. So then we looked at the spontaneous background in our TK6 line as well as in HAP1 line. And as you can see, TK6 cells have very low mutation rate, which makes them very advantageous for our studies. And if we look at what kind of signatures, this is a multiplication basically of the previous slide, one can see that this signature is practically all SBS40. And as opposed to TK6, HAP1 cells actually, as well as human-induced pluripotent cells, RP1 cells, and many other adherent cell lines which are used in culture, the background signature is dominated by oxidation signature SBS18, which is almost absent in TK6. So then we wanted to ask the question, so what is causing this mutational background in TK6? So we sequenced more than 50 cell lines that we got, and we were looking for the lines which display lower mutation rate than wild type. And the one line which had a particularly lower mutation rate was REF7 knockout. And we saw the similar effect with REF1 knockout. So what is REF7? REF7 is a regulatory subunit of DNA polymerase zeta, so REF3 is the catalytic subunit, REF7 is the regulatory subunit, and REF1 is a protein which recruits polymerase zeta to DNA. So then we decided that we need more mutations to analyze the Pol zeta deficiency signature, so we grew the cells for a very long time. We started growing them at Christmas and we harvested them in May. So we had enough mutations, and indeed we again see that in REF1 and REF7 the mutation rate is lower than in the wild type, but also the mutational signature changes. So basically all SNVs are decreased compared to wild type, except C2As and C2Ts. And if you look at C2As, then now we see signature SBS18 raising its ugly head, which was completely masked before by SBS40, which was dominant in the wild type, TK6. So Then if we look at the proportion of different signatures in the background of Rev7 knockout, we see that SBS40 is practically gone. So which basically illustrates that polymerase zeta is responsible for the SBS signature in wild-type TK6, as well as probably in many mammalian species. So when I presented this talk last month in Singapore, I was asked a question, so let's knock out Rev7 in human genome and we will have much healthier and longer lives. So unfortunately, this is unrealistic expectation because when you knock out Rev7, the cells gets very sick and they accumulate high number of structural variants, predominantly large deletions. And the reason why it is so is because translation polymerases, they try to overcome the replication blocks which necessitate the switch from replicative polymerase to translation synthesis polymerase. So they rescue cells from very dangerous situation when there is a replication block. But the payoff for this rescue is the high mutation rate. So they are very necessary, but unfortunately they have this side effect. I was talking about until now about mutants which have lower mutation rate than wild type. On another end of the spectrum, there are mutants which have much higher mutation rate than the wild type, and these are mismatch repair deficient mutants. So originally, when people were studying mismatch repair deficient cancers, in particular colon cancers, they derived 7 different SBS signatures from them. But this is an illustration of why it is important to study experimental signatures. When people were trying to analyze experimental mismatch repair deficient signatures from yeast, birds, worms, and humans, the pattern was essentially identical in all organisms. And this pattern was a combination of the 7 computationally derived signatures. So as I was saying before, if you use only bioinformatics, it's impossible to know when to stop deriving signatures. And experimental approach actually corrects it and shows that in fact there is only one mismatch repair deficient signature in all organisms. And the use of mismatch repair deficient mutant actually has another very useful application. When we made double knockouts of different DNA repair genes with mismatch repair gene, MSH2, we see the change of mutational pattern in at least 3 cases. So basically, inactivation of mismatch repair reveals these cryptic signatures which would otherwise be corrected by mismatch repair. Now I'm moving on to the second part of my talk, which is the analysis of genotoxic agent, temozolomide. Temozolomide is a methylating agent which is used in treatment of glioblastomas. So it introduces several methylation adducts on DNA, but the most toxic one is O6-methylguanine. And the reason why it's so toxic is because it can pair not only with cytosine but also with thymine, and that would cause C2T substitutions in mutational signature. But that's only a minor problem. The much bigger problem is that the mismatch repair system was designed in the course of evolution to correct replication errors. So it always works only on the daughter strand, on the newly synthesized strand, and therefore it would not correct the original O6-methylguanine lesion, but launch this futile cycle of excising the thymine which pairs with O6-methylguanine, which can then again be substituted with another thymine. And this ongoing cycle of futile repair will then pose problems for approaching replication forks, which will result in double-strand breaks and cell death. That's why it's so toxic. So temozolomide induces also a number of other adducts. Normally, O6-methylguanine is directly reversed in the wild-type cell by the action of methylguanine DNA methyltransferase. This enzyme makes the cells very resistant to temozolomide, and therefore only MGMT-deficient tumors are treated with temozolomide. It practically doesn't make any sense to treat MGMT wild-type tumors with this drug. They would be very resistant. Among other adducts, I would like to mention N3-methyladenine. which is a replication blocker, which is normally repaired by base excision repair, but also can be repaired by NAR and bypassed by translation synthesis polymerases. So this is from Phillips and Nick Zainal cell paper. What the authors did, they were determining mutational signatures which are obtained in wild-type human-induced pluripotent stem cells, so as wild-type cell as it can be, after treatment with different mutagenic agents. And surprisingly, what they saw is that methylating agents like MMS, MNNG are not mutagenic at all, although they kill the cells. And temozolomide causes a rather small number of mutations, and these mutations are not C2T, as would be expected from O6-methylguanine, but rather T2C. And to try to understand why this is happening, we employed HAP1 cells, which are MGMT+, like human-induced pluripotent stem cells. And what we see is that there is a huge heterogeneity in the amount and kind of mutations which are induced in MGMT-positive cells after temozolomide treatment. So some clones accumulate hardly any mutations, and they all fit into the same pattern which they saw with human-induced pluripotent stem cells. And these are most likely clones which express high levels of MGMT. And then there are other clones which accumulate lots of mutations, and these mutations are of completely different kind. They are C2T mutations. And these mutations, these clones are most likely MGMT low. So MGMT gene is very heterogeneously expressed in the body. So even in mouse, in the same mouse, there could be cells which express 100-fold different levels of MGMT. And so that's probably what we see in these subclonal populations. And so we tested temozolomide sensitivity of a large array of DNA repair gene knockout mutants from Takeda's collection and the ones that we generated our cells, and this is the summary of what we see. So the most sensitive lines are Fanconi anemia deficient mutants. And the reason for this is most likely because Fanconi anemia pathway protects replication forks which are getting stalled when they encounter the futile cycle of mismatch repair. On the other hand, the most resistant lines are mismatch repair deficient lines. And this is actually one of the ways to render O6-methylguanine non-toxic. So in mismatch repair deficient cell line, there is no mismatch repair, futile cycle of mismatch repair. And this is also what's happening in the course of temozolomide therapy when the tumor becomes resistant to chemotherapy. They frequently inactivate mismatch repair. So when we look at mutations which are induced in our MGMT minus TK6 cells after temozolomide treatment, we see that yes, indeed, they are mostly C2T mutations, and the number of mutations increases as we increase the quantity, the concentration of the drug. The rate of mutations is quite similar across most of the mutants, with the exception of mismatch repair. So here I had to draw it in a different scale because these cells are resistant to the drug, and they can accumulate up to 100,000, and in the case of HAP1 cells, 200,000 mutations per cell. So C2T substitutions by themselves are not particularly toxic. The cells can handle huge numbers of them, and what is toxic is actually O6-methylguanine launching the futile cycle of mismatch repair. However, if you look at how many mutations are induced per micromole TMZ, it's only about twice as much in mismatch repair deficient compared to wild type, and these huge numbers are simply because we can treat these cells with 500 micromolar TMZ instead of 10 or 5. So the signature which is then generated after temozolomide treatment in mismatch repair deficient cells is well known. It's called so-called signature 11, which was originally observed in tumors which were treated with temozolomide. And this signature is actually different from the signature which you would see in wild-type cells treated with TMZ. So the signature SBS11 results from combination of TMZ treatment and mismatch repair deficiency. And now I will slightly switch the gears. So what will happen now when we treat mismatch repair deficient temozolomide resistant cells with high levels of TMZ? Now, 6-methylguanine is not a major player, but the cells will be killed because of N3-methyladenine. And N3-methyladenine is preferentially repaired by base excision repair pathway, which I am showing here. So 3-methyladenine is recognized by glycosylase, by methylpurine glycosylase or alkyl adenine glycosylase, and which is the same enzyme with two different names. And then this abasic site, which is generated by glycosylate's activity, will be converted into a NIC by exonuclease called APEX, apurinic site exonuclease. And this NIC will then activate PARP1, which will start building poly-ATP ribosyl chains on itself on the neighboring proteins. And these chains will attract XRCC1 scaffold protein, which will do two things. One, it will recruit Pol beta and ligase 3, which will seal the NIC and fill the gap. And two, it will limit the activity of PARP1, which is very important because overactivation of PARP1 will kill the cells through energetic blockade. So then I was trying to think, is there any way to resensitize these temozolomide-resistant tumors to temozolomide again? And inactivation of one gene really did a nice job of it, and this is exactly the scaffold protein XRCC1. So if we knock out XRCC1 in wild-type cells, it has rather limited effect on the sensitivity because these cells are dying because of a 6-methylguanine and base excision repair is not involved in this process. But if we inactivate an XRCC1 in TMZ-resistant mutants, now there is a huge effect of resensitization. And the reason why the cells are dying is because they accumulate large deletions, which is probably due to unrepaired double-strand breaks. And then what will happen if we simply stop base excision repair from even being initiated by making a triple mutant with glycosylase? Then we see that the triple mutant with glycosylase becomes resistant to temozolomide again. And if we make a triple mutant with PARP1, then it becomes even more resistant, almost as resistant as mismatch repair deficient MSH2 line, which basically means that the role of XRCC1 in preventing overactivation of PARP1 is even more important than its role in base excision repair. And now, can we get even closer to clinical application? And the answer is yes. So there is a well-known paper in Cancer Cell published in 2021 when the authors were looking at exceptional responses to cancer treatment. And in fact, there was a glioblastoma patient who was first treated by surgery, then by radiation, and then by temozolomide. And temozolomide treatment cured that patient, and 20 years down the road, there was still no cancer detectable. And this is very unusual, exceptional response. And when the authors looked at the sequence of this tumor, it turned out that the APEX1 gene is inactivated by translocation. So this is now the second step in this base excision repair pathway. And for a long time, it was believed that it's not possible to knock out APEX1 in human lines unless you inactivate p53. But with the CRISPR era, it became obvious that that's not the case. So just 2 years ago, there was a paper when they knocked it out in hypertriploid cells, and they see that actually APEX1 just by itself causes very small increase in sensitivity to methylating agents like MMS. And as of 2023, actually APEX1 was knocked out in HAP1, we knocked it out in TK6, and from what I understand, it's also possible to knock out in RP1 cells. And what we see in TK6 is also that it only modestly increases the sensitivity of the wild-type cells, but now unfortunately I don't have the graph because my research officer showed me the plates only on Friday, but the result when we inactivate APEX1 in MSH2 is almost identical to what we see in MSH2 MSH2 XRCC1 double knockout. It basically, knocking out APEX dramatically resensitizes temozolomide-resistant cells to the drug. And then what happens if we now inactivate base excision repair in mismatch repair deficient mutants is that the N3-methyladenine now is dealt with by bypass, by translation synthesis bypass, and that changes the signature one more time. So in addition to T2Cs, the T2As accumulate, and this is a 3-time scale. So really T2A mutations take over, and this is probably the result of translation synthesis error-prone bypass. So as a conclusion, I would like to offer, I like to think of it as a layered response. So in order to dig up the mutational signatures, one has to remove several layers of DNA repair. So first layer is MGMT, second level is mismatch repair, then base excision repair and translation synthesis. And if I summarize it all, if we treat wild type MGMT positive, mismatch repair positive, base excision repair positive cells with temozolomide, what we see is a very small number of T2C mutations due to N3-methyladenine and translation synthesis. Then if we inactivate MGMT, O6-methylguanine becomes extremely toxic because it's not taken care of by MGMT. You have futile cycle of mismatch repair and CT2 mutations. Then, as the cells become resistant, if we inactivate mismatch repair, C2Ts accumulate massively, and the cells display this classical SBS11 signature. And then, if we inactivate base excision repair, these T2As, due to translation synthesis, take over T2Cs. So the ideal way of dealing with N3-methyladenine is base excision repair, which is error-free correction, but in its absence, it's bypassed by TLS and then T2A mutations. So the take-home message for students is, well, don't start repair pathway if you cannot finish, or you'll be in trouble. And a normal cell would rather die than acquire mutations. So to see mutational signatures, one has to generate DNA repair deficient mutants. So this project is, as I say, is a close collaboration between CGI and UNIST. It's building up on the project that our director of our branch, Professor Anton Gartner, started in Dundee when sequencing DNA repair mutants of worms. The bioinformatics analysis is done by a very talented student from Professor Simin Lee's lab at UNIST, Ju Huang. We are very grateful to Professor Takeda for giving us all these cell lines and to very many students and research officers who did the growing of the cells. Thank you very much.
Thank you very much, Dimitri, for this great overview of what happens at TMC-treated cells. I'm again moving to the back for some of the first questions. Please go ahead. You're here for a reason. While we wait, maybe I can ask one. So it's well known in the clinical situations that mismatch repair status and alkyl guanine transferase status are very important for temozolomide response. And you showed one example where AP1 may also have a role. So how prevalent do you think these secondary modifications, if you take mismatch repair and alkyl guanine transferase as the first line of defense, how often do you expect to see these alterations in this other pathway that determine clinical outcomes?
From what I understand, mismatch repair does build up of resistance due to mismatch repair inactivation does happen, but more like in 10% of all cases. So in other 90%, they develop resistance due to other means. But I think the bottom line is that when they develop resistance, the underlying cause of resistance is that O6-methylguanine becomes non-toxic. And then they are dying due to N3-methyladenine. So APEX is extremely rare case. So that I like to think of that if one can develop a drug that can inhibit APEX, that would be perhaps a good one. And then as I understand, and the REF1 inhibitor, so yeah, I didn't show it, but REF1 and REF3, for example, REF7 knockout also sensitizes mismatch repair deficient cells to TMZ. And REF1 inhibitors are being developed by Duke University and by a company in Seoul called D5. So that's another way of approaching it.
Yeah. Okay, other questions? Anybody? Entire floor?
PARP knockout did not show the sensitization, but how about the PARP inhibitor? Because a knockout probably the PVR still can do, but the PARP inhibitor, if you trap PARP1 at the damage, TMG damage, then may actually completely block the PVR processes. So PARP inhibitor is already clinically proven. So have you ever tried the inhibitor instead of knocking out PARP1?
In my experimental work, no, but yes, when I was trying to search the literature, there are very multi-numerous papers trying to use PARP inhibitors as temozolomide potentiating therapy. And the results were very discouraging. So in general, yes, the inhibitors which trap PARP rather than just inhibit are much better, but in practice it was very poorly reproducible, and for example, Professor Chen, who was visiting recently, said that it's essentially hopeless. One should give up on it.
Other questions?
Thanks. Can you hear me? Yeah. Yeah, thanks for this talk. It's so valuable to have the insight in terms of the combinations of the pathways together. It's really a lot of work. So I have a small question from the start. Is you mentioned the TK6 absence of signature 18, right? So I'm not sure I caught what is the basis of why you don't see signature 18 in the TK6.
So my favorite explanation for it is that this signature SBS18 was coined cell culture signature. And there was an idea that this signature appears anytime the cells are too stressed out. And it's very prominent in all adherent cell cultures that were sequenced, like in RPE, in pluripotent stem cells, in several tumor cells, and in HAP1. But then there are two cell lines which do not show it. And these are DT40 and TK6, and they are both lymphoblasts which are growing in suspension. So my favorite explanation is that the cells which adhere, they need to be trypsinized at every passage, and this trypsinization probably makes holes in them and stresses them out a lot. While these non-adherent cells growing in suspension, they're simply diluted with fresh media and they don't really display it. In fact, as I was showing, the signature is actually there. It's just extremely low numbers, and it becomes exposed only if you, for example, knock out REF7, then SBS40 is gone, and then you start seeing a little bit of SBS18 appearing.
And then the TK6s are traditionally used for like mutagenesis in toxicology for mutagenesis studies. So do you think that's do you think that this is behind? I don't know why it's particularly the TK6s are used, but
no, because they call TK6 because of thymidine kinase. So they were looking at the mutations in thymidine kinase as some kind of Ames assay for mammalian cells adapted for mutation, how we call it, mutagenicity assays. So that was the original application what they were developed for. I guess it's just, well, but yeah, it's a great line to work with, so I'm always happy to advertise it because of low background.
It's a lucky coincidence.
Okay, one last question, perhaps? If not, thank you again, Dimitri. And we'll continue for the last talk for this section from Professor Sung-Woo Cho from Biomedical Engineering in UNIST. So he has done pioneering work on genome engineering by CRISPR-Cas9, and today will tell us about some of the work to use this approach to target cancer cells.
Okay. Thank you for introducing me. and good afternoon. Actually, for the last decade, I studied on gene editing technology itself using the directed evolution or protein engineering. After coming to UNIST, I met Kyungjae, and he made me to study the precision medicine something, and currently we focused on an more translational applications using the CRISPR. So the CRISPR-Cas9 is widely used for the general biology or biotechnology. So for example, you can make the gene knock-in or gene knockout or bioimaging, anything else using the CRISPR. And also the CRISPR screen enables the real reverse genetics to identify the critical functional gene for a given phenotype. And based on this, many clinical trials are run to develop the gene therapy using the CRISPR. However, in cancer medicine, the field of CRISPR is restricted to use in like cell therapy, especially making the engineered CAR-T to enhance the efficacy of the immunotherapy. Before showing our recent result, I want to briefly introduce the history of the cancer drug development. There were big three innovations in cancer medicine. Sorry. The first is using the natural toxins to treat cancer. Most of these are derived from the bacteria, fungus, or plant, which are inhibiting the essential molecules in the cell proliferation. But the normal cells are also affected. It causes normal cell death and resulting severe side effect. The next is the targeting specific molecule, usually the protein in the cancer cells. For example, the BCR-ABL fusion protein is found in CML patients only, and it is critical to progress of CML development. Thus, if we can target the BCR-ABL protein, we can specifically target the CML cells, not affecting the normal cells. So, in fact, the Gleevec is the target the BCR-ABL protein effectively with minimal side effect, and most successful targeted tyrosine kinase inhibitor. But however, it takes almost 40 years to develop the Gleevecs, including the understanding of how BCR-ABL contribute to CML progress. Indeed, the Gleevec is great drug for the CML patient, but not effective the other cancers. The third one is currently ongoing, is immunotherapy. It is known to be very effective for circulating tumors, but the targets solid tumors by immunotherapy is still very challenging, and recent studies show that it has narrow spectrum of patient applicable. So the DNA damage, especially DNA double-strand break, are extremely harmful to the cells. For example, bacteria could be dead by only a single double-strand break, and yeast cannot tolerate the DNA double-strand break more than 5 in their genome. Using this, the traditional radiotherapy induced the genomic DNA double-strand break and DNA damage in the cancer cells. The ionizing radiation with high energy path through the genome. It causes various types of DNA damage, including the single-strand break and adducts. But mainly, we believe DNA double-strand break is believed to the induction of the cell death. However, it also induces double-strand break in the normal cells too. It can cause normal cell death and subsequently severe side effects. In summary, the DNA targeting method like the ionizing radiation for cancer treatment is effective way and difficult to be resisted by the cancer cells, but it has less specificity and also damage to the normal cells. Otherwise, protein targeting method is highly selective because it targets only the proteins found in a cancer. But target protein or mutations are required to be targeted and treated by the drugs. It is also true that the additional mutation on the target protein can cause the drug resistance. To overcome the current limitations of drug cancer treatment, we inspired from how CRISPR works in nature. When the foreign DNA challenged to the bacteria, CRISPR cleaves the non-self DNA and it blocks the bacteriophage propagation. We thought that if the CRISPR can cleave the cancer genome specifically, it could block the cancer cell proliferation. In fact, the cancer cells proliferation is not controlled, and the mutations are accumulated in a cancer cell upon the cancer progression. Especially the mutation occurred in a genome surveillance gene. The mutation would accelerate the tumorigenesis and might be kept in a cancer by the clonal expansion. At the beginning of cancer genomics, researchers focused on only the SNPs found in cancer, such as the driver mutation in tumor suppressor gene or proto-oncogenes. However, recent studies with the whole genome sequencing shows that cancer cells harbors a diverse of the type of mutations. Especially the recent study shows that the cancer cell containing a lot of 1,000 of indel variants, not only the SNP, the substitutions. So we conclude that even the mammalian cells are more tolerable for the double-strand break than microorganisms. Certain numbers of double-strand break can kill the cancer cells, as shown in the radiotherapy, and cancer cells harbors of cancer-specific mutations not found in the normal cells. So using these factors, we hypothesize that if we can target the cancer cell-specific indels using the CRISPR, then we can induce the cancer cell-specific double-strand break and subsequent the cancer cell-specific cell death. Here, the indel, the term of indel actually not essential. We just initially target the indel mutations because we needed to ensuring the cancer cell-specific targeting by the CRISPR. So we published this research in the last year, and we named this technology as a Cinderella, a cancer-specific indel attacker. The fact that many double-strand breaks by the ionizing radiation or the chelating reagent can effectively kill the cancer cells. But we wanted to know that DNA double-strand break induced by the restriction enzyme also effectively kill the cancers. Because the restriction enzyme leaves the 5' phosphate and 3' hydroxyl group, which can be repaired easily by the DNA ligase. So in order to this, we used the SIS1 restriction enzyme, which can be induced by the tamoxifen treatment. Upon the induction of SIS1 enzyme, it completely killed the cells very specifically. From this study, we roughly estimate the number of required double-strand break to kill the human cells. In the human genome, there are thousands of SIS1 target sequences, but the other NGS studies shows that only 100 of these target sequences are cleaved because the ACIS1 is sensitive to the CPC methylation. So we conclude the less than 100 double-stranded break are required to kill the cells. And next, we wanted to more precise quantitation of the double-stranded break numbers to kill the cancer cells. In order to this, we use the guide RNA targeting multiple locus in the human genome, and then we mark the cells with two different fluorescence proteins by non-targeting guide RNAs or the multiple targeting guide RNAs. And by measuring the ratio of two markers by the FACS, we can estimate cell viability quantitatively. In this experiment, the less than 30 guide RNAs, actually the 30 target sequences are sufficient to induce complete cell death in cancer cell lines. We also observed that this CRISPR induced the cell death in various cancer cell lines, including the colon cancer and breast and blood cancer cell lines. And in these cell lines, we cannot observe the significant difference across the cell lines. Actually, we tested more lots of cell lines, and only the fact we believe the cell death efficiency is affected by the transfection efficiency, actually. So next we wanted to examine the multiplexed CRISPR, not a single CRISPR targeting multiple locus. We identified that the HST116 cell line specific mutations compared to the U2OS cell line or the reference genome. So we first validate the indel sequence and specificity of this guide sequence in vitro cleavage assay. As you can see here, the guide RNAs are specifically induce the double-stranded break, the amplicon from the HST116 cell line, not from the U2OS. And we deliver these multiplexed guide RNAs to the HST116 cell lines or U2OS cell lines. These HST116 in their targeting CRISPR induce the cell death in a HST116 cell line, not affecting the viability of U2OS cell lines. In the opposite case, we deliver the U2OS specific guide RNAs. The U2OS cell lines effectively is dead, but the HST116 cell lines was not affected. So we wanted to make this method to more improved version because we thought that using 30 guide RNAs at the same time is a little bit difficult to utilize in clinical trials. So we wanted to reduce the number of the target needed to induce the cell death in cancers. So first, we designed a library containing the guide RNAs targeting multiple locus in a human genome, and we screened by the fitness screening and sequenced the guide RNA library abundance and analyzed the factors affecting the cell death. So, as you can see here, some guide RNAs having several tens of target sequences in a human genome, but many of them are not affect the cell viability. By the analysis of using the unbiased clustering or some other techniques, we figured out the factors affecting the CRISPR-induced cell death, and we finally concluded the conditions, the optimal conditions to kill the cancer cells less than 10 guide RNAs. And using the more improved algorithm, we more improve the target selections. And finally, I'm sorry I can't show actually what we did, but we surprisingly succeed in killing the cancer cells using only 4 guide RNAs in the cancers. So CRISPR targeting the HST116 cells completely induce the cell death, but not affect the HeLa cells. So the next, our major question is why the cells are too sensitive to the simultaneous double-stranded break induced by the CRISPR, because there are more than 1,000 double-stranded break in the cells during the DNA replication, but regarding the endogenous programmed double-stranded break and maybe exogenous delivered double-stranded break are different. It's only 4 double-stranded break, and the cells are very sensitive to these 4 double-stranded breaks. So we measure the double-stranded break and the target locus. When the 4 guide RNAs are delivered to the cells individually, for each site are repaired completely within 24 hours. But the 4 guide RNAs are multiplexed and co-delivered into the cells. Most of double-stranded locus are not repaired and remained until 48 hours. So we are trying to find the factors why the small numbers of double-stranded breaks are not repaired and remained in the cells. And we also found that the programmed double-stranded break, like induced by the topoisomerase inhibitor. So here, at the side, the cells dead, most of the cells dead by the apoptosis, but we observed that multiplexed CRISPR-induced cell death is combined with necrosis and apoptosis. So we are also currently trying to figure out why the cells and the cells response to the small number of simultaneous double-stranded breaks. So, by the way, we believe the most purpose of this method is this targeted DNA therapy is independent from the function of genes. So, for example, when we address the radiotherapy to the cancer, we are not trying to a very precise aiming of the radiation to the cell like the RAS genes in the nucleus in the cells. So any position actually can induce the cell damages. So we tried to subtract each guide RNAs from the set, which can induce the successful cell death. None of the guide RNAs seems very essential. So any combination of guide RNAs are sufficiently to kill the cells. And also we further designed all 4 guide RNAs are located to the intergenic regions and non-regulatory regions. Only 4 guide RNAs targeting intergenic regions are also completely induce the cell death. So again, we proved our algorithm for selecting the target sequence for efficient cell death. And although we consider the off-target effect of the CRISPR when we design the targets, there are still concerns of the off-target mutagenesis by the multiple CRISPRs. So we measure the frequency of mutations at the targeted locus by the targeted PCR and sequencing. we cannot observe any mutations induced by the CRISPR delivered. So we also tested the 4 guide RNAs effectively kill the cells in vivo xenograft without any weight loss or the observable side effect. So lastly, when the resistant clones are expanded from the drug treatment, there is no further way to treat these clones actually, and maybe the patient would be dead by the cancer's progression. So, however, we deliver the Cinderella method, multiple CRISPRs. We can target the mutations presented in these cells again and again. We just need to know the not the driver of cancers or the drug of targets. We just needed to know the cancer-specific mutations by the DNA sequencing. So, thus, we think that Cinderella method can be a powerful way to treatment the cancers with no treatment options or very severe stage. Okay. In summary, the Cinderella can induce the cancer-specific cell death by the multiple double-stranded breaks, and introducing the multiplexed DNA double-stranded breaks simultaneously very effectively kill the cells, and recurrent mutations and clonal expanded mutations can be targeted again and again. So, Cinderella is free for function of the genes, most importantly, and we are currently studying the underlying mechanisms of these damage-induced cell deaths and the development of the applications for in vivo study and clinical trials. And thank you for your attention.
Thank you very much. Thank you very much for this nice overview of all the Cinderella work. Any questions? Going to the back. Any questions? Gunnar.
Thank you for interesting topics. I thought that if the guide RNAs are only cancer specific, then maybe DSP repair pathway inhibitors might be also useful to specifically target the cancer. So have you ever tested those kind of drugs?
Yes, we try to combine the DNA break repair inhibitors and multiple CRISPRs, and it is very effective, but now it is too premature to show. But if you are interested, you can email to me. I will share and discuss later.
Thank you.
Other questions?
Yeah, so it's interesting. The 4 double-strand breaks are enough to introduce cell death. So do you think these targeted sites are keep cutting by CRISPR? So you introduced the 4 double-strand breaks, and that was enough to kill cancer cells. So do you think these 4 targeted sites will keep cutting by CRISPR-Cas9, or you introduced the double-strand break only one time?
Because when the double-strand break are repaired without mutation, the CRISPR-Cas9 can cleave the DNA again. So it is, actually I believe it is impossible. The CRISPR will cleave the DNA again and again, but as estimated by the qPCR, the DNA target sequences are remained as cleaved and not repaired precisely.
Yeah, because you're proposing this for possible therapy, I was just wondering, have you done experiments to look at what happens to benign tissues when they're targeted, or are they affected at all? Or?
Actually, we tried in a patient-derived cells in vitro and in vivo. Most of the patients are sensitive to this method, so still we tested not pretty much, but we also interested to figure the CRISPR-induced cell death resistible cells or the background genetic factors.
Maybe I have one. Do you think all sequences or all positions where you induce the breaks have an equal effect, or are there regions that are maybe highly transcribed or in compact chromatin or something that have a larger effect on Syndella versus others, or you haven't observed that? I guess your data shows it's pretty similar to what you've used so far, right?
Yeah, based on the library screening data, we analyzed the multiple factors may affect the cell death, but we only, the majority of the effect from the CRISPR guideline itself, not the context of genome or the environment of the epigenome things.
Okay. Any last questions? If not, thank you very much again for this nice presentation and for all the speakers in this session. And we'll take a 20-minute break and be back at 4:10, right? Yeah.
네, 제가 따로 할 거고요.
Okay, let's start the second session. Please have a seat. Okay, the second session will be a little bit different. Yes, in first session we talk about the basic of the precision oncology, but now we are switching the gear and we are more talking about the clinical applications. So the translational precision medicine will be the second session. And we have 3 speakers, and the first speaker is Professor Dr. Mark Rubin. He is currently Professor and Director of the Department for Biomedical Research at University of Bern, and he is also the President of the Bern Center for Precision Medicine. And before he joined the University of Bern in 1917, he was working mostly in U.S. in Boston area, and he got his BS at University of Wisconsin, and he got his MD from the Mountain Sinai School of Medicine, and he started his professor job at Columbia College of Medicine and University of Michigan, and he also worked at the Brigham and Women's Hospital of Harvard Medical School. And today he will talk about the minor intron splicing and novel vulnerabilities in cancer.
Well, thanks very much for the invitation as speaker. I'm really excited to learn more about the Institute, and it's already been a great first session, so hopefully we'll keep things going. In the introduction, it was sort of alluded that this is more clinical, but I think that one of the exciting things about precision medicine is that the difference between basic and clinical is really disappearing, and I think there's a lot of merging of both, and you'll see that, I think, in this presentation. Oh, there we go. So have disclosures. So there are 2 themes that I'm going to bring up that haven't really been discussed so far. And one is that alternate splicing plays an important role in cancer. And this is just from a review where it shows various types of alternate transcript splicing that are important in cancer and also in normal development. And then also I'm going to talk about a theme called lineage plasticity, which is a consequence of therapy, and I'll show this in a specific example, which is the context, which I'll be talking about prostate cancer. A lot of what I'm going to be talking about is probably very relevant to cancer in general, but I'm going to focus on prostate cancer as a model. That's what my laboratory studies. So in prostate cancer, just for some very quick background, androgen deprivation therapy or hormonal therapy is the main way that you treat the disease, and this has been known since the 1940s. What's changed is the effectiveness of the various anti-androgen therapies, and more recently, and this is a very complex slide as far as therapy is concerned, just to demonstrate that the therapies have improved in the advanced setting, where now, for example, PARP inhibitors, so we heard about PARP earlier, for patients who have BRCA alterations is now included in part of clinical care. On the other side, to the genomic side, we know a lot of information about what happens in early disease. So we've actually classified prostate cancer very nicely with genomic alterations. And we also have observed over numerous studies over the past few years that during the course of therapy, there are increased mutations that are only seen after therapy. And this is just a very nice study that looks at transcriptomic data. And just highlight one aspect, which is you don't have to read what the genes are, but what you can actually do is you can take the genes that have been identified with progression, and you can demonstrate that some alterations or mutations really only come up as disease progresses. And so I'm going to talk about a few things, so a resistance framework, lineage plasticity, and then the main focus is going to be on minor intron splicing, which I hope I can convey to you what that is and why we think it's important for cancer and why it might be a potential therapeutic target. So first, very briefly, what is lineage plasticity? The main concept of precision medicine, and we heard some nice examples about other targeted therapy, but the main goal is that you have an index oncogenic pathway that you target. And there are a number of mechanisms that can occur for resistance. So you can have the actual endpoint of the index target, then have feedback, you can have amplification. So in the case of prostate cancer, if you're targeting the androgen receptor, you can imagine all sorts of ways where it can become mutated, amplified, or there could be changes that increase, for example, the copy number that lead to the activation of this oncogenic pathway. But in addition to that, there are other mechanisms of resistance, and one that I want to highlight is where the tumor cells no longer are responding to the index oncogenic pathway, but now are indifferent to that and have taken on a new shape or form, and that's called lineage plasticity. And in prostate cancer, but also breast cancer and bladder cancer and other cancers, lineage plasticity is a very common mechanism of resistance. And my lab has been focusing on trying to understand the mechanism of how this actually occurs. So just to show you specifically, in prostate cancer, this is a very pink lesion. It's a local, it's an advanced disease that has been treated since a patient had local disease. And after anti-androgen therapy, the patient developed this disease. So now it's no longer pink, it's blue, and this means that you've seen a conversion from an adenocarcinoma to a neuroendocrine, very aggressive tumor. These patients usually have a survival of somewhere between 7 and 12 months. Over the past years, we've tried to understand what is the mechanism for this transdifferentiation or lineage plasticity, looking at genomics, epigenetics, transcriptomics. And I think one of the important findings, which is not surprising, and actually the same for lung cancer and other cancers, is that it tends to be a tumor that has a p53 and RB alterations. So that's a very important component to this transdifferentiation. It's more complicated than that. And as a pathologist, one of the things that's quite interesting is that you see that these tumors that go to neuroendocrine tumors can actually have a lot of features of the neuroendocrine tumor, but not look like a neuroendocrine tumor. So we have a problem where the phenotype doesn't actually always look like what we're calling it. And so there's a disconnect between the genotype, the phenotype, and we felt that there have to be other ways to help explain that. And so our group and others have been focusing on epigenetic regulation and other mechanisms of regulation. And the reason being, and why this is so important, is because patients who have androgen-sensitive disease, or disease that can be continually targeted with hormone manipulation, have a much longer survival. Once you have a neuroendocrine tumor, the survival is very short. And so this is a project that we started a number of years ago now with Rahul Kanadia's group and led by Anke Augsbach in our group in Bern. And luckily, after all this time, it led to a paper after many, many revisions. And if you're interested in looking at the paper, I'll tell you about it today, but you can go to it using that QR code. So what do we know about alternate splicing and cancer in general? We know that it's associated with cancer development. So as cancers progress, there's more alternate splicing that occurs. And there's a great interest in immunotherapy because it's felt that neoepitopes can be developed through these altered splice variants, and there's a great interest in that. Our interest is a little bit different, and I guess it's supposed to be a very clinical presentation, but this is probably more basic, which is that if you want to think about how we define an exons and introns, the typical explanation is you have to first be able to recognize what an intron is. And for those of us who haven't thought about it, it's really important to remember that there's a whole machinery that's in place to recognize an intron, and it's very important for that intron to be removed, otherwise you won't have a productive transcript to produce a protein. And so there's consensus sequences that are present on small nuclear RNAs that are basically the working machinery of identifying consensus and allowing the intron to be excised. There are also associated proteins, but it turns out that in a very small percentage of genes, they contain a very unusual intron, and it's called a minor intron. And this is what we're showing here. And these minor introns have a different consensus sequence. So that means that realistically, from around 20,000 genes, around 700 have these minor intron containing genes. And what that requires is, it requires that they have a slightly different machinery. And this is, part of the machinery is these two types of small nuclear RNAs that have catalytic component. And I'm going to be focusing on one of them, which is called U6 attack. So I'll talk about that a little bit more. Now, why that's important is that for these introns to be recognized, they have to have these special machinery for the minor intron splicing to be excised. And this is interesting because it's really been first identified probably in 1996, but the mechanisms are really only more recently being explored as to how they function and why they're actually there. So there's still a lot of open questions. And this figure here shows, I'm just focused on the U6 attack, that in order for the intron to be excised at the very last step, U6 attack and U4 attack, so two small nuclear RNAs have to come together and they have to excise the intron. And why that's important is because this is a rate limiting factor in excising the minor intron and also then in producing a productive transcript that can be then translated into a protein. And from basic science work, so not only from cancer, but from basic science work, it's very important because in development, the removal of these minor introns is critical, and there are a number of developmental diseases that are associated with mutations in these minor intron-containing genes or in the minor intron machinery. And what's really important is that what we've seen is that there's some regulation going on as how abundant the U6 attack is, because that's absolutely critical for normal cells. So it can't be too high and it can't be too low. It has to be the right amount. And this is a highly evolutionary conserved mechanism, so it's seen in a large variety of eukaryotic cells. So the theory is that in normal, you have a pretty low level of this U6 attack as well as other components. And then what happens is that the intron does not get excised, and these minor intron-containing genes then become degraded. In a stressful situation, the U6 attack activity is higher, and we think MAP kinase might be one mechanism for overexpressing U6 attack. And this then allows for the excision of this minor intron from a gene that contains this one minor intron, and then protein is produced. So why is that important? Well, for a number of reasons, it's important in development. It's highly conserved, and it tends to be enriched in certain conditions, and particularly in cancer, and so I'll tell you a little bit about that. But one other, I think, important concept is that these minor intron-containing genes are probably expressed in different settings. So I'm going to focus on cancer in a different context. So if we ask the question very simply, if you take minor intron-containing genes and use only these 700 genes to stratify in an unsupervised way cancer, you would find that it very nicely stratifies cancer. And in part of the revision of this paper, the reviewer asked us to expand this to over 2,000 different tumors and to try to demonstrate in an unsupervised way that it predicted the different cancer types better than randomly taking genes. And it performs very well. So if you have 100% of the analysis using minor intron-containing genes, it performs significantly better, suggesting that there probably is a very context-specific expression of these minor intron-containing genes. The other thing that's important, and this is now focusing on prostate cancer, is that if you were to ask this is just for prostate, but it's also true for other cancers, along the lines of progression, which minor intron genes are being expressed, you can see that the minor intron genes alone can, using this principal component analysis, can differentiate between benign prostate tissue cancer and advanced cancer. And so I think it's an important concept that it seems to be very context-specific and probably important. And then if you were to query which genes are associated with these minor intron-containing genes at a protein level, and you do a protein-protein interaction analysis, which was done with our collaborators at Yale, Mark Gerstein's group, they took genes that were known to be associated with prostate cancer, and they found that the relationship between minor intron genes and these so-called prostate cancer genes were at the first order. So it's highly significant and at the first order. So they seem to be very associated with cancer. But I would also point out that many of these genes, for example, BRCA and MYC, are certainly not prostate specific. So this is a consortium, a collection of cancer genes, but not prostate cancer specific genes, suggesting that minor intron containing genes are very important for oncogenesis, and they're very closely related to cancer-causing genes. Now, one of the first questions we asked was, do we see the activity or the increased expression of the various components of the minor intron machinery upregulated along the lines of cancer? And what we've done is we've taken cell lines and also patient-derived organoids, and we've demonstrated that as you progress, you see higher levels of U6 attack present in these cell lines. We can demonstrate in situ, using in situ hybridization, that you have higher levels of the U6 attack in metastatic samples or in primary cancers that will metastasize. So we have samples that we've collected for many years, and we know that those patients are going to have metastases. And if we look at primary cancers that we know haven't metastasized, they have much lower levels. So suggesting that this is associated with a more aggressive disease. We've looked functionally by using a assay where we can detect minor and major intron splicing using a vector assay and putting them into different cell lines. And what we've seen is that the activity of minor intron splicing increases with progression, but the activity of major splicing is unchanged. So this is actually important, suggesting it's more specific with disease progression. And if we look at transcriptomic data from a number of these cell line models, we also can see that all of the minor intron-containing genes are also overexpressed, as seen here for these different examples. Doesn't matter what the names are, but these are examples of advanced prostate cancer. So one of the concepts we have is that with disease progression after therapy, we start seeing an increase in this lineage plasticity, which I told you about, and it seems to be very closely associated with activity, increased activity of minor intron splicing. We've explored different conditions. So in prostate cancer, it's extremely important to think about what happens in the setting of removing and adding androgens. And when we've done that, we see changes that are expected for what would happen in minor intron splicing being regulated by androgen but not major splicing. So it seems that minor intron splicing, if you add androgens, can be increased in expression, and if you remove it, it'll go down. But nothing really seems to happen with major splicing activity. And then we also looked at MAP kinase activity. And this is, for us, it was important because in the advanced models of lineage plasticity, where we have neuroendocrine disease, we wanted to try to understand what could regulate that type of tumor different from the androgen-sensitive tumor. And we saw that MAP kinase signaling, when stimulated, as shown here, only increases minor intron splicing in the neuroendocrine tumors, these advanced tumors that have undergone lineage plasticity, and we don't see those changes with the major splicing. We can also reverse that by knocking down MAP kinase signaling. So we think MAP kinase signaling and androgen regulation could be two ways in prostate cancer that minor intron splicing is regulated. And that's of great interest because there have been some recent papers suggesting this JAK-STAT signaling is important, which is upstream of the MAP kinase activity. So what happens if we block U6 attack in cancer cells? So the first thing that we see globally is that if you block it, you have retention of these minor introns. And so why that's important is it means that you're going from cells that have proteins that are being produced by the minor intron machinery. If you knock it down with U6 attack SI, you start to now increase the number of transcripts that have the introns that haven't been removed, and they undergo degradation. And there's specific types of splicing that's occurring. It's mostly cryptic, but I'll leave that for later if people are interested. When we've done proteomics, you can see a very dramatic change, and I think the thing that I would highlight is if you look at proteomics and transcriptomics together, when you knock down U6 attack, what you see is changes in genes of great interest like RB, but also AR and genes associated with DNA repair. But when we then take a look at some of the go terms that are associated with knocking down U6 attack, we see cell division and DNA repair. DNA repair is very dramatic, and there's a really nice story that's developing about how we think we can co-target U6 attack and DNA repair in breast and prostate cancer and potentially other cancers. But I'm going to focus a little bit now on the cell cycle aspect. So in cell cycle, when we look specifically at what happens with cell cycle in the cells when you knock down U6 attack, we see a G1 arrest, which is quite interesting. And that's seen very consistently in all of the model systems that we've seen. Now, the one thing that I think is particularly interesting is if you think about cell cycle, and I think we heard a little bit of discussion before about proliferation, the E2F factors, transcription factors, are very important in cell cycle regulation, particularly for G1 arrest. And what is emerging from this is that if you look at the repressors and the activators, what's interesting, I'm not sure if it's, let's see, it's on the next slide. What's interesting is that most all of the E2F factors are minor intron containing genes. So why is that interesting? Well, just statistically, if you have 750 genes, it's about 4% likelihood that you're going to have one of them as a minor intron containing gene. What is the likelihood that you have 6 of 8 genes being a minor intron containing gene? It's not very likely. So this is, we think that's telling us something about biology, that minor intron genes, and this is just related now to cell cycle, we think also that there's a role in DNA damage, but they're very specifically developed over time that they can regulate stressful situations. And we think this represents an important vulnerability. I won't show the data now, but we have data on cell lines that demonstrate that the activators and repressors, the two types of E2F factors, are affected by U6 knockdown, U6 attack knockdown. Now, we saw in earlier presentations treating cells, so we have to ask the question, if you treat normal prostate and cancer cells with U6 attack, what happens? And in this co-culture study, we can show that the normal cells are not affected, but the tumor cells are affected by knocking down U6 attack. And this is just showing some examples of fibroblasts, and fibroblasts are not affected, and tumor cells are affected by knocking down U6 attack. And here we've combined it with some of the common anti-androgen therapies, but also epigenetic therapies, to demonstrate that U6 attack in cancer cells that are resistant to standard hormonal therapies are very sensitive to knocking down U6 attack. So this is a first types of studies in vitro that are suggesting that we want to obviously explore this more for therapeutic reasons. We've extended this now also to a wide variety of patient-derived organoids. So these are patient samples that have been maintained in culture, and we show also great sensitivity to most of the advanced tumors that we've studied. Now, the last concept I just want to bring up is something called poison peptides. So the idea is that normally I said that if U6 attack and the minor spliceosome is not working, the intron is maintained, it's retained, and it undergoes degradation. But it's also possible that the protein is formed, but it's a aberrant protein, or not the normal protein, when the intron is excised. And so just to give you more clarity on that, the idea is that you have a gene that contains major introns, but one minor intron. And if that minor intron is not excised, you have two fates. One is degradation, or the other is something else. And so the something else that we think is that you could potentially have dominant negative peptides that form that have a mechanism of regulation. I'll just give you one example that we've found. So unfortunately it's cut off here, but REST is a gene that's very important in neural development. So REST acts as a brakes for neural development, and in a normal setting where you don't want neural activity to occur, so the neural fate is turned off, you have REST expressed. It binds the DNA and prevents neural development, the neural fate. But it turns out that there's a cryptic intron that is, when not excised, leads to an isoform called REST4. And REST4 acts as a homodimer and prevents REST from binding DNA and allows for neural fate development. And so this is an important concept. As we look at that, what is really happening is that the REST4 has lost this DNA binding domain. And so, and as you can see here, this homodimer allows us then to see that you have expression of neuron-specific genes. So when we look at the actual tumor cells, what we see is that in the adenocarcinomas, but not the neuroendocrine tumors that have we have very low REST expression, as expected. So you expect low REST expression in a tumor that has neuroendocrine differentiation. That's also true for lung cancers that have undergone neural differentiation. When we look at REST4 in these tumors, we see it's higher. Now, what happens if we manipulate this? And so we can actually demonstrate that if you knock down a REST with U6 attack, what happens is that REST goes down, and as demonstrated here. But the question is that what's actually happening? So what's actually happening is that we're seeing with U6 attack the cryptic intron which is normally maintained, goes down. So actually knocking down REST4. And when we look carefully, we can see that REST4 is decreasing with U6 attack. So if we wanted to take a tumor cell that's neuroendocrine as a tumor and try to revert it, one thought would be that we would knock down U6 attack, and that would help remove this dominant negative peptide that may be driving their neuroendocrine differentiation. That's just one example. We have other examples now, and now we're going to try to look at this globally to see whether these peptides that are formed with the lack of U6 of minor intron excision may lead to a poison peptide, as I've illustrated. So the concept really here is that we have a very small portion of the genome that have introns that are called minor introns. So the typical intron would be one of many, but it's only usually one, and that requires a specific machinery for splicing. And when it's not spliced, we can have two fates. We can have a fate where it's degraded and you get a lack of protein, but you could also, if you knock it down, have a poison peptide. So we think that manipulating U6 attack might be a way that we could manipulate and make a new vulnerability for cancer cells. So losing some of that. So just going back to the beginning, lineage plasticity we think is very important in advanced cancers and how they get around the therapy. And we think that there are a number of factors that play an important role, genomic, which we've identified, but also epigenetic, which I haven't spoken about today. And then we think an additional feature could be splicing and how that occurs in minor intron splicing. I've tried to illustrate a little bit today. We continue to work on that. And so this is the group. Unfortunately, some of the names are cut off a bit. From our group, Anka Ausbach, who's been really leading this project, and our close collaborator, Rahul Kanadia at the University of Connecticut, as well as others here. So thank you for your attention, and happy to take questions. So it's definitely a little bit different than DNA repair. So.
Yeah. Yes, it is very different. And we have plenty of time for questions. Any questions from students groups?
Thank you for your talk, Professor Rubin. I have just one naive question. Was there any study that changed the normal intron to the minor intron and affected any difference?
I'm sorry, I didn't hear that.
I was wondering whether any previous study have tried to change the normal intron into the minor intron and see the differences.
Oh, okay, that's a great. So could you change the sequence of a major minor? So that's interesting. I think you probably could. I mean, it's not. But I would just remind you that in both normal and tumor, there are minor and major introns. So it would be sort of a complex thing. So I'm not sure. It's just technically, I guess you could do it. I don't know if people have done that. Yeah. But I think just one concept I just want to bring up is that there are knockout models. So if you knock out the ability for minor introns to be excised, so by knocking out any of the small nuclear RNAs, it actually is lethal. So what really are looking at is trying to decrease, like with the rheostat, turn it down but not turn it off. Yeah.
Thanks for your presentation. I'm a postdoc in Dr. Gardner's lab, and I wondered when the tissue generates metastases, the newly generated cancer cells share the same MIG profile? It's like an expansion of this MIG in the newly generated cancer cells?
So whether they have the same. Yeah, so I think the tumor cells so far, so if there's heterogeneity, we don't know about it yet. And so let me explain one problem technically, which is that most sequencing that's done excludes the small nuclear RNA from sequencing. So if you look at sequencing data, you almost can't find it. So we've actually developed a new protocol for sequencing, which includes keeping the small nuclear RNA in the sequencing, and we're also doing long read sequencing to help better align it. And so the answer is probably there is heterogeneity. I mean, in cancer we generally see heterogeneity might parallel what we see genomically, but it hasn't really been thoroughly investigated. So it's a great question. It's something that is still being explored. But technically, if you were to, after this talk, go and try to look for the expression levels, it's going to be very difficult unless you do very, very deep sequencing or use specific protocols. That's why it's mostly been missed. Yeah.
Thank you for your interesting talk. I'm PhD student in Orlando's lab. So I'm just curious, have you just tested minor intron splicing in prostate cancer? Have you ever tested those effects in other lung cancer or breast cancer?
Yeah, so in the paper, we show some data on lung cancer, breast cancer. I'm not presenting today on breast cancer, but we have some very nice data in breast cancer where BRCA status and U6 TAC knockdown are important. So that's more, it'll come out hopefully soon in 2024.
Yes, thank you for your talk. You mentioned the difference in expression patterns in the minor intron genes between the healthy and the cancer prostate tissue. But I was wondering whether you know or whether you've perhaps studied if there's also a difference between the benign hyperplastic prostate tissue and healthy prostate tissue and cancerous tissue.
Okay, I didn't get that.
So the hyperplastic benign prostate, if there's also a difference there in minor intron gene expression.
Okay, so whether hyperplastic prostate has higher expression, or is that the question?
Yeah, the benign hyperplastic. So the
fibroblasts or? Oh, benign. So BPH.
Yeah, BPH. Yes.
Okay. So I'm an expert in prostate cancer, and I don't like the term BPH that much because it's a clinical term. So what I would suggest is that if you look at benign prostate and areas of proliferation, we don't see high levels of U6 attack. So, but BPH per se is a clinical definition. It's not really, there's no association. So I'm not exactly sure what that is. But for benign and for localized cancer, we see pretty low levels of U6 attack, and that's what I showed in that one in situ hybridization slide.
Okay, thank you.
Yeah.
Any other questions from the student? No? Okay, any? Yeah.
Thanks, Mark. It was great to see it all put together. I was a little bit confused about the increased gene expression data after, at the end, you really emphasized this, the transcription of the incompletely removed portion, but then at the beginning, when you have the original gene expression data, if I understood correctly, you have an increase in gene expression of the genes with the minor intron targets in cells that have defects in the splicing or cells that are very good at it.
Yeah,
I'm missing the connection between the increased gene expression.
So I'm not going to go back to all the slides, but I'll just restate it because this is a point that is difficult. We're actually trying to make a little film so we can actually show this. But I think what's important is we see an increase in the, if you look, for example, or actually I think I have, okay, so if you look at whether it's U6 attack or any of the other small nuclear RNAs associated with the minor intron machinery, you see an increase with progression. So increase in transcripts for those. We're using a special protocol to detect that. Now, if you then go to just tumor samples, you'll see the same thing for aggressive tumors. And then if you look specifically for retention of the minor intron, you'll see that in advanced cancers because for cancers that have where we've used U6 attack. So when we use U6 attack, we can demonstrate in these tumors that they retain the intron because we've turned off the minor intron splicing machinery. So those are two elements. And then I'm not sure if that addresses what you're saying or
yeah, and so in that initial gene expression that you see, you're kind of including both types of transcripts in that
right.
analysis. And then another minor question is, you looked at genes that were commonly mutated in prostate cancers, which are generally mutated in a lot of different cancers, and then you did an analysis of that they have a tendency to have. Well, actually, I think you did an analysis of other genes that tended to have minor intron splice sites, but I was wondering about those, like the actual commonly mutated genes. If are those, do those have an underrepresentation of the minor intron sites?
Yeah, so not many. BRCA does. But most of the genes that are the main oncogenes don't necessarily have minor intron-containing genes. But there's a database. So now when I hear a talk, I usually look in the database to see if it's a minor intron-containing gene. PARP is a minor intron-containing gene. It's interesting. And so you can show regulation. So one of the concepts we think would be really cool is if you find a gene of interest and you see that it's regulated through minor introns and that it leads to this, for example, poison peptide, which is the case with REST. So for example, probably not for this crowd, but for the prostate world, REST is really exciting because people have been focusing on REST as a potential driver of neuroendocrine differentiation. So the fact that we've identified that most of us haven't looked at the isoforms, and so just the fact that you have this REST4 that comes up during progression suggests that there might be other types of methods. So there are some genes, but as I point out, it's only 0.4. It's a very small percentage of the genome, so we're not seeing that. But what we are seeing is if you look at Go terms, the effect is on, things are important like cell cycle and DNA repair. Yeah.
So this is a really intriguing mechanism of what happens in the tumor cells. So what is the minor intron splicing needed for in non-cancer cells?
Yeah.
So surely it hasn't evolved to drive cancer, right? So where is it really important otherwise?
Yeah, so again, I guess having sort of a favorite answer. So my favorite answer is, because this really intrigues me, is that there has to be some mechanism in stress where you have to react quickly, and you have to have a repertoire of genes that react quickly. So the fact that it's DNA repair, cell cycle, and some others, I think is suggesting that MAP kinase plays an important role in these stressful situations. So I think that's been known, and it's intriguing that cancer cells potentially could hijack, as sort of a good term, hijack that for taking advantage of the machinery to allow them to grow. But it is odd. I mean, why do you have this small group of minor introns? And we don't like the term minor intron because it suggests that it's not important. But yeah, that's what it is.
And so one other aspect of stress response to DNA damage, and of course in other situations as well, is difference in translation. So how translation is regulated. Do you think there's a connection between the regulation of splicing and regulation of translation, or are these two independent stress mechanisms?
I don't know if they can be entirely dissociated. So I think we're actually, so as I pointed out, one of the issues is that in order to really measure this, we have to have a new tool. So we have a new tool, new pipeline, and we're doing the long reads to try to really demonstrate what are the different isoforms that are occurring, and then also we're doing the mass spec so we can put that all together. In the paper we showed some mass spec data, but most of the techniques were sort of traditional, so I think we need to extend it with the long read technology so we can map it correctly.
So, if I understand correctly, what you're arguing is that genes which are expressed in highly proliferating cells have a relatively high propensity of having these small introns. Hence, that becomes more relevant as you go into tumor or late-stage tumor, at least in the pancreatic example, and then interfering with this mode of splicing would sort of more specifically target more proliferating cells. Is that sort of the overall rationale?
Well, I don't know rationale, but I think that's observations. I mean, observation is that, and it's not directly related to proliferation. So it's activity of cell cycle, but not necessarily just proliferation.
So it is driving the cell cycle.
Yeah. The other important point is, I think it's not just proliferation, although that comes up as the top feature. So I think there's certain hallmarks of cancer that come up as being important. I think proliferation, DNA repair, and we have, as I mentioned, I think there's very compelling data that DNA repair plays an important role here as well.
And then you would expect that you can proliferate naturally or in a cancer more efficiently if those small introns are spliced more efficiently.
Yeah.
So based on that rationale, would you have maybe mutations in U11, U12, and U4 promoters that sort of drive?
Yeah.
Or in whatever transcription factors that repress them, the other way around?
Yeah, I can't show right now because I don't have it ready, but it's really cool because if you look at how you do sequencing, there's a little blip that's cut out of the gel. Usually you don't include it in the sequencing, and that's exactly where we need to look to answer that question. So we are doing exactly that. So one of the things that we've proposed is to do extensive sequencing to look for mutations in the machinery, because we agree with you. I think whether it's polymorphisms or mutations, it might be important to look at how patients or tumors respond differently. So I think that is something that we're investigating. There are reported mutations in major intron splicing genes that have been seen, and they've been associated with developmental diseases, neural development, and there are some examples in cancer that are suggested in major but not minor. So to date we don't have an answer, but we suspect. Yeah.
Thank you.
Okay, any other questions? Okay, then please join me. Thank you.
Thanks for your attention.
Okay, the next talk will be the Professor Kyung-Gon Kim. He is associate research professor at Department of Medicine at University of Busan. He's also associate professor in Asan Medical Center. He will talk about the application of proteomics on clinical specimen for discovery of drug target and biomarker. Thank you.
Yeah, good afternoon, everyone. So it is a long talk, and some of you looks tired. But yeah, this talk is some kind of some introductive, some slides. So please, keep in mind that I want to tell you that the home-taking message is that proteomics is just like this one, and we can use or you can apply our proteomics in your some research. Yeah, please just let me start with the first one. Okay, wow, it's automatically. So today I will talk about some kind of application of proteomics on clinical sample, and its purpose is to discover the biomarkers and some drug target. So I think most of you are familiar with the proteomics. So I will talk about what is proteomics at first, then I will move why the proteomics is irreversible or irreplaceable, just like as genomics. And we will introduce how we can perform the proteomics with some application in clinical sample. Okay, let's move to the first topic. What is proteomics? So the presenters just explained many things about genomics and transcriptomics, and now it's proteomics time. So, okay, so sorry. So proteomics is the dictionary terminology is that the entire complement of protein that is or that can be expressed by a cell or tissue or organism, just like the living system. Its terminology is combined by just like genome at that time because there's no definition in proteome. So we just use this genome and protein all together. And from the very early of the 2000, proteomics is very expanded well. And so the proteomics, I can say that the scientific area which measures what kind of protein there is, how much protein change in a living system with a kind of some systematic biology. Wow, it's so fast. So sorry. So why the proteomics is irreversible, irreplaceable? So I think you are very familiar with that kind of some pictures, the central dogma. So most of us, the former presenter, talk about the DNA and RNA. So in terms of some protein coding gene, so there's 20,000 protein human genes are mapped, and the mutation or methylation or some kind of modification of gene is just studied. And now the complexity of this molecule is very higher than DNA in RNA because, as you the former the present tell, there is some the alternative splicing, so its complexity is increased. How about protein? The protein, there is some more the complication is in existing in protein. Sorry. So because there is some post-translation modification, just like the phosphorylation or acylation or glycosylation, and more averagely, single proteins are interact with the other proteins, just at least 5 to 50 proteins to make the functional links. So I want to tell you that DNA and RNA protein, so there's just some block, the building block, ATGC or AUGC, but in protein, we need some 20 amino acid. And in terms of the rule, DNA, they have the information very stable, and RNA, they have transient information or some kind of very some partial function. But I want to tell you that in terms of the function, the protein have only function, and majorly the proteins are working in cell to make this function. So the protein is very complicated to understand. Even though there is a single protein, this protein, when this protein comes up in cell surface, its function is changing. And also, if there is some binding partners, the secondary structure just changing and have some minor function, or they change some binding partners. And also, whether they have some complex with by homotetramer or homotrimer, they have different function. And when they change their function, when they make some different partners, just when they make some partners with the DNA, wow, it moved well. And when they meet with some the ribosome protein, they have the different functions. So we expressed this characteristic of the protein as a moonlighting protein because they have many diversity in function. So protein is very critical to understand the biology. From the embryo to the adult butterfly, there is dramatical phenotype changing. But during this morphogenesis, the genetic information is very static. We can explain that this change of the phenotype as a change of the gene product, just like RNA or protein. But think about that. Only protein have the function. So if we explain what happened during this morphogenesis, we need to know what proteins change and how much proteins change in each step. So I want to tell you that protein is also important to understand the disease. They are identical twins. That means they share the identical genome. But 20 years after, one side has some neurofibrinolysis NF, but the other one still there's no disease. I can tell you that. Can you distinguish two people by genetic information? We may, but mostly they share the identical genome. How can we explain what happened in their body? So sometimes he may got the same disease someday, but we don't know what is triggering factors, and we cannot monitor when he got the same disease with some brothers. So sometimes mRNA can tell us something different with some proteins. This is mRNA data with the protein data for some 12 kinds of the human tissue. So x-axis is mRNA expression level, and y-axis is a protein expression level. So some average R-square is 0.4. It's good still. But how we can explain this green dot? In green dot, the mRNA expression is increased, but protein expression level is decreased. And this blue dot, mRNA expression level is decreased, but protein expression level is increased. So we need to explain what happened to this part. So mostly this happened because there is some discrepancy in the half-life of the mRNA and the protein inside the living system. And also there is some copy number difference between mRNA and protein. So if we see some cross-sectional time, specific time, and we just put out some mRNA protein, we cannot, if we compare together, there's some discrepancy. So, but if we see the mRNA expression level and protein expression level longitudinal way, it can explain something. But these two proteomes are very different. Proteome and the transcriptome looks very different. So there are some three kinds of the clusters. This cluster of the protein, they have some very similar expression level with the mRNA and the protein. But how about this one? When mRNA decreases, the protein is increased. So if we see some cross-sectional data of the transcriptome data or the proteomics data, there might be some misunderstanding. But in terms of some function, so we need to focusing on the proteome. Also, mRNA is also important. So protein is very useful because it have been the biomarkers for the diagnosis or monitoring. We cannot monitoring some disease with the genetic data because it is static. Because when you visit the clinic, when you pull out some your blood or when you biopsy your tissue, they check what protein is expressed in your tissue or in your blood. At the same time, most of the drug target is protein, not DNA, not RNA. But mostly, recently, there is some siRNA or some kinds of some different type of the drug, but mostly 99% of the drug is targeting protein, just like some phosphatase or kinase or some GPCR. So in terms of the drug development or biomarkers, so I can tell proteomics is also important. So how we can do proteomics? So compared to some traditional biochemistry, we just generate the massive data, just like genomics and transcriptomics, using the mass spec with the S chromatography. So LC chromatography, so it is a technology to separate the peptide or protein from the complex mixture, just like tissue or blood or cell lysate. The mass spec is some kind of technology to ionize or fragmentation of some peptide ion or protein ion, and we can detect some peptide or protein ions, and we reconstruct the sequence database, sequence information from the protein sequence database. And also, sequence database is from some Human Genome Project. So what can we do using mass spec? We can identify and we can quantify the proteins in the living system at the same time. We can identify the post-translation modification, such like some hydroxylation or ubiquitination or phosphorylation, acetylation, methylation, smolylation. It is the only way to identify some PTM on the protein and how much BTM or what amino acid residue are involved in PTM, there's only way to research that things on the mass spec. So we can do the very same things, just like the transcriptomics or genomics, just like we can do the cluster analysis, and we can identify specific some cluster of a protein which involved some phenotype. We can identify the interactome, or we can do some gene ontology, or we can do the PTM mapping with the proteomics data. And this can be done using this open source database. So in the student, you can use this site. Well, there's a good tutorial and good data, some data which you can use, but they are all free. And for the mass spec, it is very essential to proteomics. And nowadays, we can use the tabletop mass and tabletop LC. And 20 years ago, when I started the proteomics, we needed to run the two-dimensional gel, and we just put out some gel to do the in-gel digestion, but you don't have to do. There's some tabletop LC mass, and we can use the liquid handler, just like the genomics. And now the data process software is very freely used. You can download it on your website, and you can run in your laptop computers. So it is very easy to do, start. And nowadays, mass spec is very useful and very changed to very fast and more quantitation. So SPD is a sample per day. So nowadays, we can analyze some 200 samples and with some 4,000 proteins per day. Or if you use some 30 samples per day, we can identify 6,000 proteins with some easy manner, with the high throughput manners. So it is well platform shape. Traditionally, we just analyze the serum or plasma, but we can analyze the spanurine or colonoscopy biopsy and FFP slide tissue. And today I will talk about this one more. And we can analyze what proteins in there in exosome, or we can do some single cell proteomics now. And in this day, I'm working on the single cell proteomics. So using the single cell passing some capillary, we can isolate the single cell, and we can put this single cell on this plate, and we can digest, and we can make a peptide, and we can do some proteomics things with this one. So we do use some Selenium-1, this platform, to isolate it and prepare the cell. And using some 40 SPD, that means 40 samples per day, we can identify the 6K proteins in a day with some 40 samples. It's well developed. This is the simple example for the single cell proteomics. They are HeLa cell. HeLa and with a different size. This size is some 18 micrometers, and this cell is 27 and 30 micrometers diameters. So we just isolated this HeLa cell. So we think that the HeLa cell line, they might be the same in proteomics. But yeah, they look very similar in proteomics, proteome numbers, but they show the different characteristics even though they are single cell. So small size of the cell showed some different proteome rather than compared to some larger cell. So for the larger cell, they have some mitochondrial or metabolic or stress response proteins expressed well, but the smaller HeLa cell, they showed some proteins in RNA binding or splicing or transcription. That means even though they are same single cell line, but they show the different characters in protein, even though they are cell line. So that's why we need some single cell proteomics. So, and also we are moving to some spatial proteomics using the FFP slide. So we just captured some using the laser microsection, where we collect the very specific, the single cell resolutions, the FFP slide with the 10 micrometer thickness. And we prepare the proteins in using 96 well plate, and we can do the same proteomics things with the computational manners. Okay, so I will introduce some kind of example in clinical samples. First one is a glioblastoma. It's just a brain tumor. So when you visit clinics, you can get some this kind of imaging exam or some blood test, or you can get some tissue biopsy to get some tissue exam. So FFP is a formaldehyde fixed paraffin embedded tissue. So it is a very useful data because they have spatial pathological information and some outcome data together. So traditionally, FFP is a very useful resource for the DNA or RNA analysis, but now it is very useful for the proteomics. The proteomics is very hard to do because in the process of making FFP, there's a fixation or some kind of dehydration or infiltration. They are not good for the proteomics analysis. But nowadays, we can capture some tumor specific area using the laser capture micro dissection. We don't have to keep some frozen tissue, which costs a lot. And we can use, because they are all stored in pathological department or biobank. So there is some good publication because when they compare some FFP and the frozen tissue together, deep frozen tissue together, it is good correlated well. And there is some good pathology to make a T-cross-link, the cross-link by some ship base caused by formaldehyde. Using some pH or temperature or buffer, we can decross-link again. So there's a lot of some good methodology about to do some FFP proteomics. So let's move to the glioblastoma. So there's no cause about the glioblastoma. There's some genetic background altogether. And this glioblastoma consists of the 15%, and the overall survival is only 12 months. So when you got some glioblastoma, the survival duration is at least 1 year. And the 5-year overall survival is 3%. Diagnosis is only some CT, MRI, and some tissue exam. And the treatment is surgery and the radiotherapy and the chemotherapy with the temozolomide, as you told in previous lectures. But temozolomide, it is very easy to make a resistance. So nowadays they used Avastin, bevacizumab. They just blocked the new angiogenesis some factors. So it just a very next generation treatise. So it is very critical to determine the proper treatment strategy in the early time. So our question is that how we can distinguish the patient-people treatment, because even though we got the bevacizumab treatment, this patient showed some efficacy. So tumor is just shrink, but this patient, after some treatment of bevacizumab, it just refused the other side, and there is some metastasis or some kind of recurrence again. So we cannot distinguish this one using a genetic data yet. But I want to know that after some surgery, so by examining this tissue, how we can distinguish whether this is he is bevacizumab responder or non-responders. So we did some quantitative proteomics analysis, and fortunately we just find that non-responder and the good responder show the difference in proteomics, and also we can identify some clusters of protein which increase in the non-responder. and the increase in the good respond well, just like these proteins. This is averaging, and there is a 7 patient, and there is a 7 patient of the no response, and there is a 7 patient with a response. Some proteins is very exclusively expressed, and when we do the IPA analysis, they are involving in cell-to-cell signaling and some pathway to the resistance. So necrosis and the proliferation of the neural cell and invasion of the neural cell, different proteins are involved in each patient. And our aim is that we can apply this protein panel to some multiplexing IHC after the surgery to determine that the proper treatment strategy. Also, we are selecting the druggable target for the GBM. For some Avastin non-responder, we can select some kinase or E3 ligase, and one of them is very effective to reduce some Avastin resistance in non-clinical animal model. So our strategy is that, so after surgery tissue on the protein analysis within the 2 hours, we just classify the bevacizumab non-responder or responders. Then if he is some responders, we can do some in-time treatment well. But if he is some non-responders, we can identify, designate he is some non-responders, so we can discover the new drug target, and we are working on this stage. So let's move to the second example. The one is the triple-negative breast cancers. So triple-negative breast cancers, they are very mesenchymal-like and stem-like one, and it is very hard to difficult to do. But in clinically, when they visit some clinic, we do some anthracycline neoadjuvant chemotherapy. Then we do some surgery, and when we examine in pathology analysis, we just classify whether she is a non-responder or non-responders. So we cannot distinguish before this diagnosis step. So we have some core needle biopsy FF tissue sample in the stage of some diagnosis. So we want to map this outcome data onto some sample on some diagnosis well. So we do some protein analysis using very tiny piece of some FFP sample. So we just use 20 of the PCR or 37 of the non-PCR. PCR is pathologically completely responders. So it is very similar to, we can distinguish 2 groups with some very tiny piece of the proteome data with the FFP. And also we can identify some specific pathway about some non-resistancy. Then we are working on some knockout mouse model making, and we are working on non-clinical data analysis. Third one is cholangiocarcinoma. Cholangiocarcinoma is a kind of some very hard to treat some carcinoma. So the 5 years relative survival in USA is below 10%. And this paper, this TEMP randomized trial, they show that capecitabine and gemcitabine plus the cisplatin, they showed no difference in outcome. So we just used this kind of sample again. So this is some paraffin block. So the pathologists just mapped some pure tumor area. We do the proteomics analysis, and we just mapped this CCLE. CCLE is just cancer cell line encyclopedia. So when we put this data on our data is some blue one, and some background data is a red one. And this dot line is biliary tract cancers in CCLE. So we can identify that in our data, there is some angiogenesis feature is very strong. And also there is androgen response pathway is higher than the normal cancer tumors. Also, you can do some single cell gene set analysis using our data, protein data. So we can find that this patient showed some short survival or duration, and this patient have the long-term survival. So when we mapped some single-cell data on the tissue, this patient showed some lower proteins in the macrophage, just like this one. And yeah, there's a difference in monocyte here, and they showed some different single-cell features, even though they are some global proteome data. And also we identified some TFS correlated protein, just like this one, and we can identify some high-risk group and low-risk group DP, differentially expressed protein, to identify some tumor data. This protein, they have some capstatin treatment, some prediction power, not in gemcitabine and cisplatin treatment. But as you told you, in using some clinical data, they cannot distinguish this treatment, but using the proteome data, we can distinguish some capstatin, the responders sample patient well. So this is a pancreatic neuroendocrine tumor tissue. This tumor is related to these cystic jobs. We just use the normal tissue and the FFP, PNET tissue altogether, and we identify some good proteins pathway, just like myogenesis and some mesenchymal transition. And we identify some druggable target, 37 kinase or 23 channel protein and cell membrane receptor proteins, which shows that increase more than 2 times in peanut tissue and extracellular location using the bioinformatics. So, and one of them, we just do some possible proteome together. We identify peanut tissue specific proteome data, and we mapped some kinase. We cannot say when the kinase abundance increase, the kinase activity is not increased. So, but we can say if there is a post-peptide increase, it's correlated kinase activity is high. So using the post-peptide information, we can map some 6 kinase, and one of them, when we block this kinase with siRNA, it works well to some tumors model in the mouse model. So we are working on some cell surface proteome and the cell surface glycoproteome using the biotinylation now. So our target is some discovery of some CAR-T target, which not exist in normal tissue, but only exist in peanut tissue well. So last is the ovarian cancers. Ovarian cancer, we just identify some BRCA1/2 mutation carriers, some specific protein only increase in ovarian cancers. To use that, we just use the 40 microliters of blood plasma, and we identified some specific cluster which increase in the ovarian cancers with some BRCA1/2 mutation. So we identify many proteins in blood, and we identify 24 significant proteins, and just like Oncotype DX, we want to make some model to distinguish the ovarian cancer patient and the normal patient with the BRCA1/2 mutation. So we just used some machine learning approach, and using the 5 proteins, we can distinguish these two groups, BRCA1/2 mutation carrier with ovarian cancer and no ovarian cancer yet. It is the same with another machine learning instrument. So our aim is that if she got some BRCA1/2 mutation carrier, so we can do some blood test every month, and if the score is higher, we can do some next intensive diagnosis. So now we are moving to some FFP tissue with very early. It's very tiny, just like some rice, single rice. So we do the same thing with the proteomics. Early tumor and some normal tissue show the very distinctive proteome, and we also identify drug target which is increased in tumors, and also we identified some new subtype of ovarian cancers. Now we are moving to some different type of some cancers with follicular type of thyroid tumor and some HCC and MPNST and Gaucher disease. This is a working some project. So yeah, in some medical centers, there's some good facility and good infra for the quantitative proteomics well. So if you need any some collaboration or any some help, please let me know. And we are working on some prediction, monitoring, and diagnosis biomarker discovery with a single cell proteomics. And thank you for your attention.
Okay, any questions? No questions from student? Okay.
They have to pay a beer for me now, the students. Anyhow, just thanks for the interesting talk. Just a couple of sort of very basic questions. So if I give you 1 microgram of extract, how many proteins can you resolve these days with ease, and how many can you resolve when you really try very hard?
Okay, very practical question. So in terms of the single cell, so its quantity of protein is 25 picogram. So if you give me some 1 microgram, so we can identify at least, I'm sure, at least 5,000 proteins in there. So we can identify the 5,000, yeah, based on the single cell data. So yeah.
So you can identify 5,000. How many can you reliably quantify?
Yeah.
Because you know the dynamic range of proteins is 10 high 6, 10 high 7.
Yeah, good question. So it depends on your some characteristic of your sample matrix. So if you have some plasma or some kind of some sample, there's just some dynamic, the abundance issue. So at the plasma, there's some, I think 70% of protein will be quantified. But if you send me some cell line data sample with some there's no dynamic range issue, yeah, 95% protein can be quantified.
With 1 microgram, so to say.
Yeah.
So that is, and you know, if I had asked you the same question 4 years ago, what would have your answer been?
10 years.
5 years ago.
5 years ago, so if you give me 1 microgram, so no, my answer is I cannot take your sample. Yeah. But yeah, 5 years, yeah, there's some big difference or big development in the mass spec or proteomics.
Thank you.
You're welcome.
Thank you for showing us all the things you can do with proteomics at Asan Medical Center. So you have shown us a lot of interesting research results. So how far away, or are you already using any of these for routine clinical diagnosis at this point?
Yeah, good question. So if we have discovered some some target, just biomarkers for the specific disease, we need to get some approval of the KFDA. So, but if I were in USA, there is some lab development assay, LDT, and also there's a CLIA lab system. So even though there's no approval of some FDA, we can use that assay. We can use that biomarkers for some clinical purpose. But in Korea, very, very conservative. So we need at least 5,000 of sample data to get some approval. We need more time, we need more some sample, or we need more money. So it takes a lot of some effort.
So you need to show the reliability of your assays in a larger number of patients before you can use it to make a clinical prediction.
Right.
Okay.
It's correct.
Yeah.
Unfortunately.
So a lot of work ahead, I guess.
Yes. So some of the companies just moved to the USA or Hungary or European country to get some approval of a CLIA lab. And for during the CLIA lab performance, they can collect some data
they can deposit some example or reference data. Then they move to come back to Korea again to get some FDA approval.
Is there like in Korea, if you have like a disease that is, where patients have a very small chance of survival, and maybe you have a last straw of hope in one of those assays where you find something that you might target, even in that case, are you not allowed to use it in the clinic?
Yes, I can do personally, and I can. There's just some urgent some system to apply this kind of some assay. So there's just we can get some approval easily for the. But only in case of emergency, we can do that.
Thank you.
You're welcome.
Great to see all the technology. So I just a technical question. So I know with the single-cell RNA-seq, there's a lot of care that's be taken into the pre-analytic parts of the preparation. How much variability do you see with the patient sample, and how is it going to be prepared so that it's reliable?
Yeah, great question. So 1 month ago, there is some World HUPO, Human Proteome Organization conference. At the time, there was a single-cell proteomics session. And one guy just presented some single-cell proteomics, and at the very same cell, they did some RNA-seq. So they just compare. Can you guess what R-square for some single-cell proteomics and single-cell RNA? Right, it's 0.02. It's very low. So if we see some bulk RNA-seq and bulk protein data together, the correlation is 0.4. It's good. But if we see the single-cell proteome and single-cell RNA data, this correlation is very low. That means we need to do more things in single-cell proteomics and single-cell RNA. So yeah. There's some publication for that.
And is there any selection for type of proteins that tend to be represented, or is it?
Yeah, so put up some single-cell proteomics, they just measure, they just detect as much as they can. So there's some detectable protein, so there's no panel or there's no target for the single-cell proteomics. And RNA-seq, there's a lot of some RNA gene set, but there's some limited number of protein list. If they compare, R-square is low. Yeah.
Okay.
Just for the cancer analysis with proteomics, isn't there a little bit of naivety if I take a tumor which is 1 centimeter cubic, I might have 1 billion cells, and each of the cells has, let's say, 10,000 mutations. That essentially means that every nucleotide of the genome is mutated 3,000 times. I.e., before you even start your treatment, you already have resistant cells. And even if they are very rare and not detectable by RNA or proteomics, once you change selections, they will prevail.
Yeah, good question, but I cannot answer that question now. But it's a very good question. It's very challenging for me. Yeah. Yeah, there is some heterogeneity in cell, and in each cell there is some information, high level of information about mutation or mutation correlated to some outcomes. But in single-cell proteomics or in proteomics, I think they are not ready to explain these things yet. But someday, just like 5 years ago, 5 years later, there may be some answer for that. I hope so. Sorry. Yeah.
Okay, thank you for the great talk. Okay, let's move on. And today's last talk, but not the least. Okay, Professor Tokun Kim, he's from the Department of Pathology at Asan Medical Center. He will give a presentation about utilization of clinical NGS data. Yeah, he's preparing. Yeah, ready. Thank you.
Thank you. Thank you for inviting me here. My name is Dr. Kim from the Department of Pathology at Assam Medical Center. Today, I will be presenting on the utilization of clinical NGS data. My presentation focuses on introducing the system developed at our hospital for precision medicine. Precision medicine refers to the provision of objective and reproducible healthcare services by quantifying and integrating various patient-derived information, such as genomic data, in addition to clinical information. Among the various components of precision medicine, I will be presenting the establishment of a system for utilizing clinical genomic data for precision medicine at Assam Medical Center. Among the various systems utilized at Assam Medical Center, those related to precision medicine are depicted in the diagram here. Today I will specifically focus on systems related to genomics. I would like to present two major genomic-related systems here and here. The first one pertains to the Genomic Laboratory Information System, GLIS, which is used for the collection of genomic data. The process of clinical NGS testing begins with a test order, sample preparation, sequencing, data analysis, and variant review, ultimately culminating in a generation of clinical report. GLIS system is a one-stop system designed to streamline and facilitate this entire process. The primary users of the GLIS system include medical technicians and data analysts, pathologists, and physicians. We utilize the core functionality of LIMS system to manage all stages of genomic testing. Through integration with the EMR system, the test order information and clinical NGS report are automatically linked. During variant review, we have the capability to reconfirm aspects like tumor cellularity by interfacing with the digital pathology system. In the context of copy number estimation, it is highly sensitive to tumor cellularity. When utilizing the observed variant early frequency and digital pathology, accurate copy number estimation can be archived efficiently. Genomic data collected through the GLIS system is configured to be loaded into the Clinical Research Data Warehouse, CRDW here, for research purpose. The second genomic-related system is the Precision Medicine System, PMS, designed for the utilization of this data in precision medicine application. Issues arose when the data was used independently. As for clinical information, here, it is hard to explain clinical significance of genomic information. As for genomic data, it is hard to search related clinical trial. As for specimen information, sample usage rate is very low without genomic information. We have developed a PMS system as a system for leveraging genomic data. This system addresses the challenge of fragmented utilization of clinical information, genomic data, clinical trial information, and specimen data, which has been a significant issue affecting the usability of the data. Through the system's implementation, we were able to develop PMS system that facilitate the integrate and linkage of diverse data sets. The PMS system has been developed with two main purposes. One is clinical usage, and the other is research use. In the clinical context, patient information is displayed in a non-anonymized format. In the clinical PMS system, integration with the EMR system allows for patient search. Users can click on the precision medicine icon after searching for a patient. And this action displays the detailed information of that patient in a time series format, like here. The research-oriented PMS system has been configured as a separate system accessible only to in-house users. It can be utilized for various medical conditions such as cancer and asthma and neurological disorder and etc. For cancer disease, we classify them based on a primary site and morphology, and we have mapped these classification to MSKCC's Oncotree, like this. This allows to select specific cancer subtype and create code for analysis. After selecting the cancer subtype, researchers can build specific cohort for their studies by setting detailed filters such as TNM stage and SEER data, age, treatment status, genomic information, and specimen availability through the dashboard interface. After generating the cohort for patients who have undergone NGS testing, researchers can use the OncoPrint screen to visualize the overall mutation frequency, providing a comprehensive view of mutation within the cohort. Once the cohort is established, patient identifiable information is anonymized, allowing researchers to access a patient view screen similar to the clinical view. Regarding specimen and sample-related information, researchers can determine when samples were collected and whether any residual samples are available in the biorepository core for further research use. For drug treatment and radiation treatment, the treatment duration and prescription details are displayed like this. Furthermore, it is possible to review the result of pathological and radiological examination like this. In our case, pathology test is there's no result, but the imaging test we have the result, and we can display the result in the monitor. For individual patient mutation, we present them in the table format similar to SBio portal. And if there is a knowledge base like CIVIC available, we provide detailed information in a pop-up window for reference. if there are patients within the research cohort who have samples stored in a PRC, the system displays the number of samples that are available for retrieval. Additionally, the system offers functionality to request these samples through a formal request process. To overcome the limitation of previously shown dashboard or patient view with restricted filtering options, we have recently implemented a feature that allows for more granular conditions to be included as filtering criteria when creating cohorts. This includes incorporating event-based relationship, such as patients who undergo surgery within 30 days after cancer diagnosis to further refine the filtering conditions. The PMS system was developed with the primary purpose of confirming the feasibility of research before obtain IRB approval. Once a suitable research cohort has been constructed, researchers can use this system to request the extraction of the necessary data from the CRDW after receiving IRB approval. To build the system described above, digital formation of patient-derived data is essential for each system. At our hospital, the digital transformation of traditional pathology test results, such as immunohistochemistry and PCR, is already in progress. When these results are integrated into the PMS system, we anticipate that it will create significant synergies and enhance the overall capabilities of the system. Thank you. If you have any questions, please send me an email to here.
Okay, any question?
So I'm a pathologist. I really appreciate that.
Thank you.
It's great. So one question that I'm sure you're thinking about is, have you thought about how you're going to integrate AI approaches into this system?
Yeah, we have a plan to the kind of manner, but nowadays we just plan to the digital transformations first, and then we can transit to the use AI system. Yeah.
Any other questions?
So if I understand correctly, these data will then be, patients will be able to view their data as well, right? And so how, I'm sort of wondering how you think about how patients deal with the data. You could imagine that some people are more literate in understanding the data, and for others it may be a little harder to know what the data means. So what does Asan Medical Center do to help the patients understand and use their database, these databases?
Yeah, we also plan to explain the whole treatment courses to the patient using this kind of systems. For instance, if we use patient view mode, the patient see the whole treatment and the whole the examinations, so they can easily understand their status. Thank you.
You know, when you generate this data, how does this approach generally fit into the Korean health systems where hospitals are not necessarily sharing their data with other hospitals? So, you know, recently my wife went in the first hospital, had one diagnosis, then the second one said the first one are wrong, and well, they were wrong as well. Altogether, she was treated very well, but if she would be a research subject, the hospital not speaking to each others, it would be virtually worthless.
Yeah. So the data sharing is it's complicated in Korea because the patient data sharing is illegal in the Korea, so we cannot share the patient-derived data.
But then why are you doing research in Korea if you can't, in an anonymized way, search data? I mean, I recently encountered, you know, as a reviewer, a paper where everyone would ask for the raw data, and they told me they can't share it. So that's actually meaningless, it's worthless, it's just a waste of time and money.
Yeah.
Actually, as far as I know, Korean government tried to develop a platform where they can share the patient data, but it didn't go well. There's a lot of the things going on.
Just to comment, so, because one of my other roles is precision medicine, and so in Switzerland, I think this is a credible example, and in Switzerland we're trying to set up, the same problems that you describe are universal. I mean, it's always a problem about sharing data, but I think what's going to happen is as evidence-based studies move forward and we see that you have to share the data, I think it will, so I think we have to be optimistic that it will make sense that you share the data for a number of reasons, and this is occurring, I think. But it's breaking old traditions of provincial approaches, but I don't think it's entirely a waste. So.
Yeah. I guess again, I want to say that, you know, I'm much happier with the medical system here than in the UK, but nevertheless, you know, they generally have just one health system, one database. There are 250,000 sequences accessible, and it might weigh at the end that this is the way to go. Also, you know, in the Korean system, so it might be, you know, lots of lobbying which is required to more generally share and be allowed to share the data. Because otherwise you essentially just exclude yourself from the international community.
Yeah, in clinical genomics data, we do not share the in public, but the genomic the research purpose, it can share to the public.
Okay.
Yeah.
So I am interested in how many cells do you analyze from one tumor biopsy for the clinical, for the basic experiment. So some researchers generate organoid to detect unique mutation signatures, or we isolate clonal cell population to detect unique mutation signatures. But if you collect cancer tissues, there are many cells and it may be difficult to detect unique mutation signatures.
Yeah.
So how many cells do you usually analyze?
Yeah, unfortunately, we don't use the live cells, like cell lines. We just use FFP samples from biopsy and the surgery tissues. So we can dissect the tissues and then get the DNA. And if we can, if we have enough DNA amount, we can sequence the patient tissues. So
do you always adjust the concentration of genomic DNA?
yeah, we do not calculate the number of cells.
Thank you.
Okay, any other questions?
Okay, so I will not ask some questions, but I add some comments of the data sharing in hospitals. The first, there are two major issues in data sharing in hospital. The first one is the de-identification. So we should not infer the patient individualized information from the records. So if we think of some ICD-10 or RX norm or other codes to clarify some medical cause or claims or treatment, it would be okay. But in genomic data, I think that it would be easily inferred from the genomic data to who is the patient from the genomic data. So, and also there is the other major issue is there is no common data model. So in hospitals, there are common data models to share the data from among the hospitals. But in genomic area, in my knowledge, there is no common data model or the platform or not platform but cause to share the genomic data. Even there is some information of genomic data. So even there is some movement to share the information or data from the hospitals, but I think that it would be more difficult to share based on the genomic data than other clinical data.
Thank you for the good comment.
Yeah. Any further questions or comments? No? Okay, let's thank to all the speakers for this session. Okay, thank you for this great symposium, and we will have dinner after the session, so you are all invited. So anyone has time, you can stay and have dinner together with us. Okay, thank you. Food is ready already. Actually, it's in conference room 2. If you go to the conference room 2, there will be a food and you can bring the food here and enjoy your food.